How are we all doing? Good? Great. All right. So we're going to continue the lecture series, and the next lecture is going to be on, again, a more continuation of the basics of electrochemical biosensing devices. Yesterday, I did mention to you, at the heart of the biosensing device is a bioreceptor. So today, we're going to look a little bit more on the bioreceptors. We're going to do a lot of recap today just to kind of consolidate on what we learned yesterday. And I'm going to focus a lot today on one of the bioreceptors, which I think is going to be a future for tomorrow, right? It's being used quite a lot, and it's got a lot of potential. And I'm hoping, and I think my prediction is that this is a bioreceptor which will be doing electrochemistry fundamentals and understanding the electrode miniaturization process, focusing mainly on the bioreceptors and how the bioreceptors can be tapped into to exploit the sensitivity. And then there's also going to be a lecture after this that will be mainly on epidermal. So I've moved the lectures around because tomorrow I want you really to be very fresh, to give you one of the best lectures that I think is going to be here, which is going to be combining bioreceptors together to get more extraordinary sensitivity, getting that ultra sensitive detection that you normally expect from your biosensing devices. I do appreciate I have a wide audience here. Some of you are not working directly on biosensors, but as I mentioned yesterday, biosensing technologies are enabling technologies. You can extend these biosensors into your area of application, whether you're working in the area of forensic sciences, environmental sciences, healthcare sciences, whatever it is, you can definitely extend the applications there as well. So the lecture is, as I said, this is the learning objectives. We're going to be understanding electrochemical biosensors again. We're going to be looking at the different types of sensors, how they are fabricated, the transduction principles. Once again, we already covered a little bit yesterday. We're going to consolidate on that, and then we're going to look at some real world applications here as well. Suggested reading, this is one of the favorite books that I have, which is on analytical electrochemistry by Joe Wang. Fantastic book. There's also a paper which was one of my first papers on the use of subdermal devices, wherein we actually got the subdermal devices investigated on a microfluidic device. Microfluidics, as you all know, is where you have miniaturized channels which are used for manipulation of the flow of liquids. You can use that for having multiple reservoirs, mixing them together. So this is a nice paper that was published, I think, back in 2012. So it's a good reading as well. So I would suggest reading these two papers, and they will be made available through the organizers in a zip file where you have all the collection of all the papers. So just a quick recap. Yesterday we saw what are chemical sensors and how they become biosensors. We saw that a chemical sensor is mainly a device that would transform chemical information. Mainly we're looking at concentration. That's what we're mainly interested. In some cases we also look at the activity as well. So for example, when we look at kinases, CDKs, we are looking at not just the concentration of, but we're looking at the activity, and we measure the activity as well. But the most general example that's given is that a chemical sensor is a device that would transform chemical information, mainly concentration, of a specific sample component into an analytically useful signal, which could be in the form of a visual signal which you see as a reading on your device, or which could be an alarm. For example, if you have an oxygen depletion sensor here, which is giving you a warning that the oxygen levels are dropping down. And then, as I mentioned yesterday, your chemical sensors would have two basic components, and these components are normally connected in a series. You would have what we call as a molecular recognition element, or a receptor, which is combined to a physicochemical transducer, and the physicochemical transducer would then convert it into a useful analytical signal there. Now this receptor becomes a bioreceptor, and when this becomes a bioreceptor, it becomes a biosensor. Right? A biosensor is a sensor which would normally have a recognition element which involves or which utilizes biochemical mechanisms. That's the example of a biosensor. So we saw examples, I gave you, we went into depth, I think we're going to be covering that again, but we saw examples of how enzymes could be used for making biosensors, how antibodies could be used for making biosensors. We are basically exploiting the specificity of these bioreceptors to get your biosensors of interest. What exactly is a biosensor? As I said, again, it's an analytical device which has got a biological recognition and signal transduction element, and it measures a signal proportional to the analyte. That's the important thing here, that your biosensing device should give you interesting information or useful information which is on the concentration, and this concentration has to be directly proportional, or the current values that you measure should be directly proportional to the analyte concentration that you're measuring here. That's what makes it very important. And the common targets could be You've seen examples of glucose. You could have stress hormones such as cortisol, or you could have cytokines, as I mentioned, CDKs, examples of cytokines as well. IL-6 we saw yesterday was another example of cytokines. I showed you this slide yesterday as well. I said how we have different samples here. These are your different samples on your left hand side, and then how you could use the transducers, and your transducers which are involved in the signal transduction would have your bioreceptors, which could be nucleic acids, cells, antibodies, enzymes. These are your bioreceptors, and then you can have devices, or you can have electrical interfaces such as your field effect transistor devices that we saw. Somebody asked a question about yesterday. Nanowires, I said to you that these are very fine structures which have got a very high surface to volume ratio, and therefore very useful and give you very good sensitive detection there. You could also have nanoparticles. Gold nanoparticles and silver nanoparticles have been used. You might have heard about this technique called as surface enhanced Raman spectroscopy, right? That's where you use cracked gold, cracked silver, which gives you ultra sensitive detection there, but that's an optical detection method. That's why we're not going to be discussing too much on that, because we are focusing mainly on electrochemical biosensing methods. And you can also have electrodes. This is normally people who work in the lab who do cyclic voltammetry and things like that have already got used to this particular setup there. So this is your signal transduction. This is your signal transduction is done. Once this is done, then it's transferred to something that's what we call as an external circuit, which normally has a signal amplifier to amplify the signal to get a measurable current there, and a signal processing which can make sense, do the conversions, and then finally you have a display system which is part of your electronic system. So these are the main the components of an electrochemical biosensors, and these here are the different types of samples. I won't go much into details. We've covered that in much detail yesterday. Again, this is a recap of what we just saw. You normally as I said, the heart of your biosensor is your bioreceptor there. The bioreceptor is what gives it the required specificity. It could be an enzyme, an antibody, an aptamer, and I've introduced this new term here, MIPS. We saw a little bit of this. This is basically molecularly imprinted polymer, right? We'll see examples of this in greater detail today. When we'll have a look at MIPS, and I'm going to be spending a lot of time today discussing about MIPS, because as I said, they have potential to be the future bioreceptors, because they allow you to, and they also call as plastic antibodies. All right, we've already seen this. We move to the next slide here. This is a different slide here. The bioreceptor types that we've basically covered, or which we have understood as part of this GIAN series, are enzymatic sensors, enzymatic bioreceptors, immunosensors will have antibodies as bioreceptors, aptasensors will have aptamers or strands of oligonucleotides, and MIPS will have molecular imprinted polymers there, right? These are the different types of bioreceptors that you will normally come across, and you can see here, this is your enzymatic bioreceptor. It works according to the lock and key model hypothesis that we have. This is what gives it the required specificity that we need for monitoring or for biosensing. This was a slide that we covered in greater detail yesterday. Everybody happy with it? Anybody wants a recap of this again? So we saw yesterday how we have different generations of glucose biosensors. We have the first generation where we are generating hydrogen peroxide, right? You don't have any mediators there. This is the first generation, but then to break this hydrogen peroxide and release electrons to be able to measure current, we are working at very high potentials, and therefore to reduce those potentials, we normally work with a mediator. Mediators work at lower potentials, and that helps you in addressing the problem of interference, right? And there's also the third generation there, where you have a direct electron transfer from the cofactor, from the enzyme cofactor to the metal surface there as well, right? So this is something that we covered yesterday, and we also discussed a little bit about the pioneering works that were done by Leland C. Clark towards the first generation, Professor Tony Cass towards the second generation, and Professor Koji Sore done towards the third generation of electrochemical or enzymatic biosensors there. Similar to the glucose biosensors, you can also have lactate sensors, which could be first generation, second generation, third generation. In fact, in the case of lactate biosensors, anybody working on lactate biosensors here? You have the third generation which is available. You have a direct electron transfer sold by a company called Lactazyme, right? If you want to work on that, you can use that enzyme, and there are papers that are coming out from Imperial College where they've used the third generation lactate biosensors, which are commercially available there. If they're commercially available, they're clearly deemed to be safe. All right, when you see the word immuno, that means you're talking about antibodies here, right? And how do you represent antibodies? They're normally represented in the form of a Y shape, right? These antibodies, as you might have already studied during your school time biology and chemistry lessons, these have got very good specificity, and the interaction between the antigen and antibody is very, very specific, right? These are normally, I mean, we've been normally the main source of your antibodies that are normally natural antibodies is animal, right? So you normally extract them out of animal. It's a very, getting the antibodies out and getting, doing the purification of these antibodies. It's a very, very expensive process. It's a very tedious process. It's got multiple steps there, and it really makes the whole process very expensive. Enzymes as well. I mean, even the enzymes, when we buy the enzymes, they are really expensive. They're not that cheap, right? Antibodies, again, they are more expensive than the enzymes. It depends on the antibody you're looking for, but the rare antibodies or rare diseases are much more expensive because it's very hard to extract them and purify them and to get them to a form which can then be used for immunosensing applications. So what you see on your slide here is an electrode material, which should be a metal surface. On the metal surface, you have a nanomaterial modification. This is something which we're going to cover tomorrow, wherein we're going to talk about combining nanomaterials and then having the enzymes or having the bioreceptors immobilized on them. What is the advantage of them? We're going to cover a lot of this tomorrow because I want to keep this very simple right now. I just want to focus on single bioreceptors in this lecture, but tomorrow we are going to be looking at something really interesting, how you can combine the bioreceptors, with nanomaterial systems, and how you can combine the bioreceptors amongst themselves to get some very interesting results there. Okay, so this is basically what we're looking here is you have your different nanomaterial modifications. This looks like a dendrimer, and this looks more like a graphene kind of thing, and this looks more like a carbon nanotube here. Right, so what you're doing here is you have your antibody which is immobilized on the surface, right? And once you've immobilized that, this particular technique is called as ELISA. Anybody heard about ELISA? It's enzyme linked immunosorbent assay, right? So what is happening here is you basically have one antibody, and then you have a target molecule, and then you have a second that comes in. So your target basically is sandwiched between two antibodies, and then this secondary antibody has basically got a fluorophore there, or an electrochemical tag there, and if it's got a fluorophore, then you can measure the changes in fluorescence, and if it's got a electrochemical tag, then you can measure the changes in the electrochemical properties that you have here. So these are typical examples of how an electrochemical immunosensor would normally work. The link is down there, and you'll be getting the slides so you can follow it up. The next example I'm going to talk about is APTA sensors, right? Aptamers, basically strands of DNA, strands of oligonucleotides, right? And we all know in nature, we know that we have the double helical structure, and this double helical structure, the two strands are held together by hydrogen bonds, right? You have the adenosine pairing with thymine and cytosine pairing with guanine, right? Cytosine pairs with guanine using how many hydrogen bonds there? 3. Well done. And adenosine pairs with thymine pairing by 2, right? So you have 2 hydrogen bonds there. Well done for remembering your school level, whatever we call it, GCSE level biology and chemistry there. Well done. So that's basically that's basically how your oligonucleotides normally bind amongst themselves. But at the same time, they're also known to bind to certain molecules, and it's based on the same principle. They use their hydrogen bond capacity, and they're able to bind with certain molecules of interest, and they are very specific as well. They give you the desired specificity. As I said, bioreceptor, heart of the biosensor, has to give you the desired specificity, and not only give you the desired specificity, also give you the desired sensitivity as well, right? These are terms that you can, specificity has to be natural, but selectivity, the sensitivity is something that we can fine tune as well, depending on what we are trying to measure and what concentrations or what's the dynamic range of the biomarker or analyte of interest is in. So this is an example of how your aptamers can be assembled on gold. Anybody heard about self-assembled monolayers? SAMs? So thiol groups, you know, alcohol groups are OH, thiol groups are SH groups, right? So if you have a molecule which is terminated with a thiol group, so if you have a molecule and then you terminate with a thiol group, the thiol groups are known to form very nice layers of self-assembled molecules, right? You can have them literally because of the, you know, the forces balance out each other. The forces of attraction and forces of repulsion, they balance out each other so nicely that they self-assemble themselves in a very nice uniform layer, right? So these are your thiol compounds, right? once you know how your thiol compounds are, you can have your oligonucleotides which have been terminated with a thiol group. So as soon as you drop your thiol groups, your solution of your thiol onto a gold surface, they will all stand on it and assemble themselves, keeping the equal distance spacing there. You don't have to do anything. This is all done naturally. You don't have to assemble the molecules one by one. It's just all done naturally. And there are some beautiful AFM pictures that you can pull out from the internet, where you can see that these molecules are standing really nicely next to each other, right? Very well assembled, very good spacing, very uniform molecules. So this is something that you can exploit, and you can use that. So what you see here is basically the S is basically your sulfur groups from the thiol, and you can see that it's standing down as self-assembled monolayers, nicely assembling on the gold surfaces there. And then this is what you have here is basically your target molecules, and you can see that what happens is when it's standing straight, as soon as it binds with something, it gets a little bit heavy, and then they start to bend downwards. So as you get the electrochemical tag closer to the surface, you're basically facilitating a much better electron transport. To have electron transport, you remember when we were talking about third generation glucose direct electron transfer, we were talking about how the electron transfer groups have to be closer to the surface, to the metal surface, so that you can have an efficient electron transport there. I mean, what is electrical conductivity? It's the flow of electrons. How do you ensure that you have a good flow of electrons? You have to ensure that your electrochemical tags are closer to the surface, so that you can have a much efficient. And there's a certain value there. If you look into papers that talk about direct electron transfer, you will see that they come up with values which are normally in the range of very few nanometers, which allow where the electron transfer takes place. If you look at glucose oxidase, if you look at the molecule of glucose oxidase, the cofactors are all quite high, sitting quite high up there. So what they do is they normally fragment it and bring it closer to the surface. So that's why your direct electron transfers are normally, direct electron transfer enzymes are normally engineered enzymes. They are fragments of the enzymes, because you try to bring the electroactive part closer to the surface, so that you have a very efficient electron transport there. All right. So that's your, did I just jump? That's your electrochemical aptar sensors. With that, now I take you to what I would say is a little bit of a case study kind of a thing on a new class of bioreceptors, which we call as molecular imprinted polymers. I'm not very enthusiastic about it just because I'm working on it personally I'm working on it myself. It's also because of the fact that these are actually, have been reported in the literature as well to be very, very sensitive as well. They can, and the reason I love them is, number one, my objective is to make low cost devices, right? If I can make something which is low cost, something which is stable, if it's something that can be made by scalable technologies, what else do I need to make low cost devices? As I said, antibodies, problem is you're deriving them from animal sources. Extraction and then purification, making it really expensive. Enzymes, you're deriving it from plants. Again, extraction and purification. This is something that can be synthesized, and you can make them as you're building your biosensors. You don't need to buy them separately. You can buy the monomer solution, and then you can polymerize it. And there are, polymerization is those of you who studied chemistry know polymerization has been going on since ages. We know about all these wonderful natural polymers that are existing in the nature, and we also know about the harmful polymers that exist, that we have created, your polythene bags and all that stuff. These are polymers that are you can choose to be in accordance to green chemistry and biocompatible as well. All right. So molecular imprinted polymers are also referred to as synthetic antibodies, because you are synthesizing them yourself in the lab, or they're also called as plastic antibodies. You can modify their properties as well, right? The main advantage is you can tailor the specificity, right? If you can take a monomer, you can mix monomers together, and you can get the desired specificity. You have a monomer which is giving you a particular shape, but it's not giving the required specificity. So you can have another monomer and another polymer around it to alter the properties, to give it the desired shape, to maintain the shape. It's very important here, because you're creating cavities, you're creating binding sites here. It's very important that you maintain that the whole binding site in its native form, right, the way you create it, right? So the advantages of molecular imprinted polymers is, number one, it gives you tailored specificity, right? You can tailor the specificity to go down to lower concentrations as well, and I'll give you examples when in the next few slides as to how we have used these molecular imprinted polymers for early diagnosis of a disease called Alzheimer's disease. Anybody knows about Alzheimer's disease here? Alzheimer's disease is a neurodegenerative disorder, right? It happens, I think 70% or 80% of dementia that happens with old age is because of neurodegeneration of the nerves, right? It's a natural process, right? But the problem is that it's becoming more and more common. Again, as I said yesterday, we are all victims of our own success, right? As we have managed to bring in technologies that have increased our longevity, and we are all living longer now. Earlier, people used to die by the time they were 65. I'm talking about 1930s, 1940s, 100 years back, right? Now, we all know that the average average age is around 80, and dementia starts around 75, 74, 75. So we are all worried about the quality of life because there's a lot of dependency in that particular thing. When people do not have good control of their nervous system, they need help there, right? That's where, so we need to be able to diagnose early, and now we have medication for management of Alzheimer's. We need early interventions. We all have the medication, but we need to know when do you start the medication. So for that, you need to be able to diagnose the disease. And actually, there are reports which have clearly suggested that if you have the right diagnostic tools, you can actually diagnose Alzheimer's disease 15 years in advance before the onset. You can look at the pattern of the neuronal degeneration that's taking place, and you can predict when it's going to start. And then if you start the therapeutic intervention at the right time, you can then ensure that it's managed in a nice way. You have a good quality of life. Right? There are ways, there are different types of studies and tests there. So coming back to these molecular imprinted polymers, so what I was trying to highlight here is that we have used these molecular imprinted polymers for our project on Alzheimer's disease to basically develop early diagnostic systems. And as I said, you can tailor the specificity, and they're highly stable. So we have a UK-Japan project. So we made our devices with molecular imprinted polymers, and we sent it by courier to Japan. And when we tested them there, they were all working very fine. So they're all very stable, right? You have, if you look at the objective should always be, as they say, the world is a family. So you should always try to think of developing devices that not only work in your own conditions, but also which can work in global conditions. You should think about continents like Africa, where you have extreme heat, the sub-Saharan heat temperatures or heat conditions. You have high heat, high humidity. Even in India, you go from region to region, the temperature changes, the humidity changes. So you should have a device that not only works in Kashmir, also works in Kanyakumari, where you have the whole extreme that you have. Not only works in Gujarat, also works in Assam. You should be able to capture. Your device should have that robustness, should have that stability, that it works in all conditions. And that's one of the things that you get with molecular imprinted polymers. That's why it happens to be one of my favorite bioreceptor. So it's highly stable, it works in robust and harsh conditions, and as I said, it's very, very cost effective. You can start making them in the lab. When you're building your biosensor, it's not about, you don't have to order it as a separate chemical. You can buy the monomer and you can polymerize it yourself. And the monomers are not that expensive. Nothing compared to the prices that you pay for enzymes. I don't know what the prices of enzymes here are for glucose oxidase, but we pay a lot of money for glucose oxidase, lactate oxidase, and if it's hydroxybutyrate, it's much more expensive. So they're very expensive. Antibodies, we don't even bother looking into antibodies because we know they're very expensive. ELISA kits are based on antibodies, and we know that it costs a lot of money. A plate for Simoa analysis, which is single molecule analysis using, which is basically a glorified form of ELISA, that's about 6,000 pounds. You can do the maths now, multiplied by 100, how much it costs to do 48 samples, right? So it's very expensive. This molecular imprinted polymer gives you a much more cost-effective solution here, right? You can use this. It's much more cost effective, and as I said, durable, and they give you a long shelf life. You can store them over a longer period of time. People who work on enzymatic sensors, where do you store your sensors? 4 degrees in the fridge. Can you imagine transporting the sensors if you were to do an experiment somewhere in Kampala or in Accra? Can you imagine how your sensors are going to work, right? So transportation is a problem. I think that's the problem we had with vaccines, the cold chain thing. Right? Anything that needs to go into cold chain, that's a problem there, because then you need extra resources to be able to maintain them. You're doing all this huge program there, and then you don't know whether your vaccine is still efficacious, whether your sensors are still working or not. So you need something which is more stable and which can withstand all the harsh conditions. So this one is, as I said, is durable and has a long shelf life. You don't have to store it in the fridge. The cartoon that you see here, there's a simulation here which is done. This is fantastic simulation, has been done by my fantastic PhD student Sudhansh Deshpande, wherein he's basically showing you, what he's showing you here is this molecule in the blue. This one is your vancomycin. It's showing how vancomycin comes and binds to this molecular imprinted polymer, which is basically a plastic antibody designed for the vancomycin molecule. This is your cavity. You can see here, it comes and nicely binds there. How this all happens, I'm going to be showing you this in the next few slides. So, how do we fabricate molecular imprinted polymers? I'm sure most of you must be keen to know that, since I've told you so many nice things about it. What you do is you start with a template molecule that you see on your screen here. This could be your target biomarker, right? This could be a drug for which you're doing therapeutic drug monitoring. This could be a large molecule. This could be a biomarker of Alzheimer's disease. This could be a biomarker of brain infection. This could be something as simple as insulin. This could be something as small as glucose molecule, right? So you have your template. So you start always with the template, because you're creating, as I said, molecular imprinted polymers. You're creating a template here. How do you create a template? You take your target molecule, your molecule of interest, right, which is basically your biomarker, and then you bring it in, you put it in a solution of your monomer here, right? What happens here? You will see that, I will show that in the next slides. Your monomers will interact with these particular template molecule according to the size and according to the charge. So in the first step, that's what happens, and that's a very quick thing that happens. And then what you start doing is you cross-link your monomer so that they then start to polymerize. Cross-linking means that you're binding your monomers together to form a much more rigid structure around your template molecule, which is your biomarker. So in this third step, you have cross-linking of your monomer to form the rigid structure, and then what you do is you eat away this template structure that you have here, and then what you end up getting is you remove the template and you get your cavities, which correspond not only according to the shape but also to the charge centers. Every molecule, if you look at large molecules, every molecules have got charge centers, right? What are these charge centers and what are the interactions I'm talking about? We'll be seeing that in the next slides. But you need to understand that you need to conserve the size, the shape, the geometry, the charge centers, all of these things together to be able to get a template into which you have your biomarker comes and fits in, right? You may have a complex mixture where you may have molecules with similar size as your template, as your biomarker. They will compete against each other. Therefore, you need to make your template very specific, and that's can only be done by not only conserving the shape but also conserving the charge centers there as well. Okay, so what are the proposed interactions when you have your interaction? So I mentioned about this step here where you have interaction with functional monomers here. What helps in stabilizing it? You have, so if you talk about a polymer, a monomer such as phenol red, which is one of the monomers that we are using in our group. And we've also managed to file some invention disclosures on these. So we have basically got this phenol red. Basically, you have the hydroxy groups. You also have the sulfonyl oxygens, which are strong hydrogen bond acceptors. So you have these the hydrogen bond formation which takes place between phenol red because of the hydroxy group and the sulfonyl oxygens that you normally have. Hydrogen bonding. Everybody knows about hydrogen bonding here, where every time you have hydrogen placed between a electronegative atom such as fluorine, nitrogen, oxygen, you will find hydrogen bonding there, right? So here, phenol red basically has got these two groups here, hydroxyl groups and the sulfonyl groups, which are so at the hydroxyl group, clearly you have the hydrogen between the oxygen and there's going to be a next electronegative atom there. Sulfonyl groups, the oxygen atoms are going to be acting as strong hydrogen bond acceptors there, right? So that's what happens in the case of sulfonyl groups here. In the case of, so this is what hydrogen bonds that, this is what comes from the monomer. And if you look at the target analyte, which in this case is vancomycin, you have multiple hydroxyl groups, you have the amine groups, you have the amide linkage. I'm doing a little bit of chemistry here just to kind of explain things, what is happening at the molecular level, right? You have the amide linkage, you have the carbonyl groups in the carboxylic acid group, and then you have the ether linkage. Yeah, just delving down deep into what's happening here, what are the proposed interactions here. So you have the interactions of the hydrogen bonds, which, is it a good thing or a bad thing? It's a good thing. It helps in stabilizing because hydrogen bond is a form of an attractive force, right? It's binding it together, right? If you have forces, you may also have other forces which will be responsible for repelling it, for destroying the structures. You will also have that. You have to find the right balance there, right? So these are the hydrogen bonds that take place. You will also have hydrophobic interactions. You will have phenol red's contribution. You have got the phenol groups, right? The more carbon content you have, the more is the hydrophobic interaction there. So there's going to be few hydrophobic interactions from the phenol reds contribution. And in case of vancomycin, you again have these alkyl groups and the aromatic rings, which will also contribute towards the hydrophobic interactions, right? So the presence of hydrophobic pockets can then lead to phenol red being lightly bound to the sequestered hydrophobic packets, right? That's the kind of interaction. And this interaction is driven by the tendency to exclude water molecules. That's an important thing that we need to look into when it comes to hydrophobic interactions. At the same time, there are also going to be another form of interactions, which are your electrostatic interactions, as the name suggests, starting with both because of the charge and the differences in pH. All right, both molecules will exhibit different properties at different pH, so you need to make sure. So you normally, when you're doing this templating thing and you're trying to create the template, we want to see what are the different parameters you're trying. What is the optimization parameters here? You'll try different pH. You will try different ionic strengths, you will try different concentrations, different ratios of your monomer to your target molecule, and see what is giving you the best combination. This is what is normally developed as part of your biosensor development thing. This is what normally people who work on biosensors, this is what they optimize to get a device which gives you the desired sensitivity. This is where I say sensitivity is something that you can play around with. You can try to make your devices more and more sensitive by making more and more sites available for binding, making more and more efficient sites, and making it more and more specific, right? This is what basically what we normally work towards optimization. So then once you've done all of that, then you move to the sensor usage. This image that you see here is basically a PCB electrode here. What we need is just 70 microliters of the drop. You wait for 30 seconds, you take a measurement. I think it's 30 minutes. So you're allowing all the binding to take place, and then you take a measurement, right? So you get a cyclic voltammogram there. So basically what you need is, there's no sample preparation required, there's no washing step required, there's no external redox probe required here to probe the electrochemical properties here. That's the advantage of using this particular polymeric system. And look here, this is the dose response curve. So what we've seen, this is what's been optimized. So I'm showing you the best result here. I'm not showing you all that hard work that's been done in the back office, which is mainly done by Sudhansh himself. I'm just showing you what the final dose response curve looks like, and I'll explain you why I like this dose response curve. Here, your MIP material here happens to be polyphenol red, right? So you can have phenol red, which is your monomeric solution. You polymerize it to polyphenol red by cross-linking it. You're getting a nice isotherm here. You get the sample matrix here is, we work directly with spiked serum samples. Where do you get your serum from? You get it from your blood samples, right? So the serum samples have been obtained from blood samples, and we've spiked them with different concentration, and you can see that the linearity is all the way up till here. This happens to be the linearity range, right? Almost till here. This linearity range is about 70 micrograms per milliliter. And actually what we are looking for, this is for your p-tau proteins. If you look at the lower limit of quantification, it's 2.7 micrograms per milliliter. Upper limit of quantification is 70 micrograms per milliliter, right? How is this useful? Now we know that the typical value, the typical mean value or the median value at which you would get a full diagnosis of p-tau proteins is around 4.8 to 6 micrograms per milliliter. Normally it's 4.8 is the cutoff, but what happens is you can have people with no AD, no Alzheimer's disease, but can have a high concentrations, because this is related to your age as well. And that's why at some point I will explain how we need to bring in the machine learning and the AI models as well, right? Because you have this whole thing, the baseline, right? So all of us have some form of neuronal degeneration. At the moment, our values are sub micrograms per milliliter concentration here. So this is basically showing you the limit of detection here. So this is the concentration, and we are getting the desired sensitivity here. All right. So this is a performance. Sorry, my apologies. I'll take my words back. This is about vancomycin. Right. I confused you. I'm sorry, for apologies for confusing you. This is about vancomycin. So vancomycin, here you're looking at micrograms per milliliter concentration. Sorry, this was picograms actually. This was for Alzheimer's. So my apologies. I'll just take this back. This is for vancomycin, and that's why we're looking at micrograms per milliliter concentration. This is the typical example which I gave you yesterday of therapeutic drug monitoring. Do people remember that? Therapeutic drug monitoring I gave you? I told you. So what happens is drugs, normally if you look at the concentration, you normally would have concentration over time. For a drug to be effective, you normally administer a drug, it goes into your body and it distributes itself to reach a peak concentration and then it drops down. So before this drops down, you administer another drug, so it goes up and then drops down, and then you administer another one here, it goes up and drops down. So what you try to do is you try to maintain this concentration level here, right? And this range at which it's normally effective is called as the therapeutic index, right? You have to get your drug concentration in the blood in that particular range for it to be effective, right? And we saw the example of antimicrobial resistance. Overuse and underuse can cause antimicrobial resistance. So it's very important that you stay in this therapeutic range, right? The therapeutic range for vancomycin happens to be between 1 to 10 micrograms per milliliter, right? And what we can see here is you have the lower limit of quantification is 2.7 micrograms, and upper limit of quantification, it happens to be 70 micrograms per milliliter. That's how these normally work, right? So it's a good example wherein you got the desired range here, just with phenol red polymer here. So that's an example of vancomycin here. I'll just make it very clear. This is vancomycin therapeutic drug monitoring in serum samples, because when I saw serum, that's when I understood that I was doing a mistake here, because you normally measure vancomycin in serum samples, not in plasma samples. In the case of Alzheimer's disease, you measure the biomarkers in plasma samples, and in this one you measure it in serum samples. So what are the main challenges in translational research when you have bioreceptors such as your molecular printer? Cost of screen printed electrodes is very high. This is a PCB electrode, right? And these ones you can get them for a price of 0.09 pounds or 9 pence, right? Screen printed electrodes, how many of you have worked with screen printed electrodes? When you go and buy them, you get a box, and that box is about what, 400 pounds, 400, 500 pounds. So you're talking each one of them costing about 5 to 10 pounds for the screen printed electrodes. And screen printed technologies are normally considered to be scalable technologies, right? We didn't find them very scalable. We found them to be more expensive. And as I said, the objective of my research group is to develop low cost diagnostics for global healthcare solutions. I can't do low cost diagnostics if I'm getting something as expensive as 5 to 10 pounds. I would prefer something which is costing me 9 pence. Even if it's made in some remote corner of the world, I would prefer to source them and I would prefer to buy them. Yeah, or probably make them in my own lab if possible. Okay, so the cost of screen printed electrodes is very high. Therefore, we introduce these PCB electrodes that you see on your screen. As you can see here, if you look at this one, you basically have got a reference electrode, you have a counter electrode. This is twice the size of the working electrode, and you have 2 working electrodes here, so that you can do multi-analyte detection. You can also do something called as redundancy. This is the term wherein you measure the same analyte but on 2 different electrodes, so that if one of them goes wrong, at least you've got a value there, and you can also take the average values there as well, right? Sometimes it's very useful there to look at the reproducibility of the device as well. I'll show you devices where I think the devices that we are going to be working, they've probably got 4 electrodes, right? So you can also have 4 electrodes. You can use them for the same analyte to get n equals 4, or you can use them for 4 different analytes. The choice is yours. N equals 4 happens to be a very good set of data there. People who work in analytical chemistry, any analytical chemists in the audience? No? You understand the value of meaning of n equals 3? Rishita, n equals 3. If you have present any data which doesn't have n equals 3, you don't have the paper accepted. You need to have n equals 3, and you need to show the error bars, the variation between the sensors as well, right? You have to show reliable, reproducible sensors, right? So having 2 of these working electrodes helps in getting that. If you have n equals 4, even better. Minimum you need is n equals 3, because if you have n equals 2, you can have 2 data points, 2 points, and you can always get a straight line between those 2 points. The other things that problems we have is you also have biofouling effects on the surface. This is where we've used highly porous gold, electrochemical interference from other compounds, and expertise you need for data analysis. This is just to show you how the data analysis is done. These are the traces you normally get from the 2 different working electrodes, and then what you can see here is this is showing you how the data is collected. This is for vancomycin, PPHR. You can see this is patient 1, just to show you how we are gearing up for the human studies, right? So the results I showed you are studies that have been done in plasma, but in vitro conditions. These are not real samples. We've taken plasma, so that's how you normally get your dose response curve in the lab, right? You get the blood is sold as a reagent in the UK. I'm sure it's sold as a reagent in India as well. So you can buy blood which has been tested for different pathogens. You can use that blood. You can spike it with different concentrations, so that then you know this concentration gives me this value, and then you can get a dose response curve, right? So this is an example where then he's getting the software ready on data analysis, and basically this is an example of a slide where we did clinical samples for n equals 12, and this is a range that we get here. As you can see here, we had patients who were given vancomycin, which is the last resort antimicrobial, but they were also given a cocktail of drugs, because before giving them vancomycin, they were given different anti-TB drugs: meropenem, piperacillin, tazobactam, isoniazid. All of this was given to them, so there would be some traces of that as well. So you would expect them to be electrochemically active and some interference from these as well. But we managed to use the right set of conditions using differential pulse voltammetry and identify the exact potential we should be working on to get to identify the peaks and find out which is the best working condition here, right? Now this result is basically, this is the value that has been predicted by the molecular imprinted polymer, and this is the value that has been predicted by LC-MS. LC-MS is liquid chromatography with combined to a mass spectrometry, right? This is normally used for, it's a standard method. It's a lab technique. It's something, if anybody who's seen a LC-MS or a GC-MS, you would see these are quite big. I mean, they're benchtop, don't get me wrong, but these are not portable devices. So what we are trying to develop are portable devices which we can carry around. We can work in any part of the world, and you can work on farm side, you can work on bedside, you can, point of care kind of devices. That's the kind of things. So these are clinical samples against which we validated our devices, and you can see beautiful correlation there, and there's a good agreement between our devices, our biosensing devices, and the LC-MS data that we've obtained here. So what's the elephant in the room? Well, MIP specificity. I mean, do we need our MIPs to be highly specific, highly selective? We need it, right? I mean, the data I showed you was showing more on the lines of micrograms per milliliter concentrations, right? That's fine for vancomycin, that's fine for this application, but there might be applications where you have to go down quite a lot. For example, Alzheimer's, when you're doing early diagnosis, you're trying to predict something 15 years before. You can imagine if you did a measurement in my blood, still far away from my 70s, there would still be some traces. It's going to be trace analysis, but we would like to have sensors which can look at my concentration and then track it and extrapolate it and tell me when do you think Alzheimer's will set in me, and then I can start my medication accordingly earlier on and things like that, right? So the specificity is still in infancy. That's what we think. They may have low specificity when it comes to molecules with similar structures. That's the problem here, right? So you've got to be very careful with the choice of molecular imprinted polymers, and there are a lot of materials which can be easily polymerized to give you the templating there, okay? And then the molecules, they may not be suitable for all drugs, because some of the drugs are highly hydrophobic. They're not soluble in water, and what I showed you right now was mainly how you are electropolymerizing them. And if you're looking into doing electrochemistry on them, it has to be aqueous based. You can't have organic solutions there, right? So some of the drugs could be hydrophobic drugs. If you have hydrophobic drugs, they may not be soluble in water, right? That's a problem there. It's not a one for all kind of a solution there, although we've tried to see how many we can do, but you will have to develop your molecular imprinted polymer template according to your biomarker, according to your template molecule, according to your biomarker molecule. It's not like you take polyphenol red, it works for everything. You will have to fine tune its properties. You'll have to change its specificity, right? And then there's also this, we are undergoing some continued research and further developments here, and it's also suggested that we combine the receptors, and we also use the dual receptor approach there as well. Sometimes that helps in enhancing the sensitivity, and this is something, please come tomorrow morning as fresh as possible, so make sure you have a good night's sleep. No partying today. We're going to have a real good lecture tomorrow on combining bioreceptors to get ultra sensitive biosensors. Yeah, okay. So looking at the conclusions based on the molecular imprinted polymer work that was done, vancomycin is a glycopeptide. It's an antibiotic, which is a last resort antibiotic. therapeutic drug monitoring is essential for vancomycin because the therapeutic range that we have is 15 to 25 micrograms per milliliter. It's high, and it varies from individual to individual. This can go down to 10, as I mentioned to you before. This can go down. So if you have something where you're trying to measure between 15 and 25 micrograms, you would want to have a dynamic range of 0 to 70 micrograms per milliliter concentrations. That's how you normally design your biosensors. So if your biosensor is giving you the range of 0 to 70 micrograms per milliliter, then you know it's something that's going to be working very well. All right, MIPS can be made out of polyphenol red. This is a new material which has not been used before for making MIPS. So it's the first time we employed these, polyphenol, or we employed phenol red monomers to make molecular imprinted polymers. And then what we have also seen is you have different types of interaction between the vancomycin and the molecular imprinted polymers. There were the hydrogen bonds, there are the hydrophobic interactions, and there is also the electrostatic interactions. The dynamic range we saw is from 0.848 to 70 micrograms per milliliter, which is very good, which is giving us the desired sensitivity. We are covering the desired dynamic range, which is fantastic, and we have validated these samples against something which is a gold standard, which is liquid chromatography and a mass spectrometry system there. All right, that was all about molecular imprinted polymers. I hope I've given you a good flavor, and I hope I've convinced you why, if you were to choose one bioreceptor to start off working with, it would be molecular imprinted polymers. Although the experiments that we're going to be doing as part of this GIAN series is going to be on an enzymatic sensor, but that's just to get you used to how you build a biosensing device. But if you were to start your research on biosensing and start making biosensing devices, my recommendation, strong recommendation, would be to use molecular imprinted polymers. You can choose your own molecular imprinted polymers, right? But go for that one and then combine it if necessary, if you need a much more higher sensitivity or a lower limit of detection. Okay, coming back now again to the biosensors. What are the different fabrication techniques? I mentioned a lot about screen printed electrodes, right? That's a scalable technology. I still don't understand if they are scalable technologies. Why are they still very expensive, right? Screen printed and inkjet, the people make out of screen printed and inkjet printing. There's also the PCB electrodes that's normally made. You also have micro molding. I gave you examples of micro molding yesterday, where I showed you how you can micro mold micro needles. I gave you the example where you can take a aluminum block and use electric discharge milling, make your pyramidal structures, and then pour in PDMS and peel it off, and then you get the reverse structures, and then you can bring in a photopolymer such as SU8, and then you can make the pyramidal structures there. Okay, you can also do electropolymerization, and you can also do nanomaterial functionalization. I think we've covered a little bit of all of this, and as part of the fabrication techniques that are involved in building a biosensing device here. We haven't covered any optical biosensors at all, right? So I'm just going to give you a little bit of more information, because we are covering the basic fundamentals for biosensing. So I think it will be nice to have this particular slide on optical biosensing. Optical biosensing could be, you can have different types of measurements. You can have one measurement where you're doing absorbance or transmittance measurements, right? This follows which law? Well done, good for remembering Beer-Lambert's law, right? This follows Beer-Lambert's law, which is a simple law which says that absorbance is a product of concentration A equals ECL, right? I think I'm sure most of you have done it, or even if you've done the engineering, you still covered Beer-Lambert's law as well. All right, so absorbance, this is your Beer-Lambert's law. Here you have a source which is basically and then you scan, you then measure the absorbed or transmitted light. Here you can get a response, which if it's an absorbance you get a peak, and if it's transmittance you normally see a dip. So you can normally see that in your FTIR spectrum, where you look at the transmittance versus wavelength there. So you have a dip there. And then you can do the analysis, and your absorbance is directly proportional to the concentration here. While with the transmittance you get more like a exponential decay kind of thing there. You have transmittance and you have the concentration going down. So that's one method of doing an optical detection method. The other one is very common, which is fluorescence. Fluorescence is normally known to be very sensitive. That's why fluorescence is very common. And as I said, now if you use in this particular case, you have an excitation wavelength, you excite your molecule, and then you'll have an emission monochromator, and then you have your intensity measured against the wavelength of emission, and then you can get your plot of intensity versus concentration. Again, there's a good correlation between intensity and concentration there, following the same principles there. All right. Anybody remembers Jablonski diagrams that we had for fluorescence, to explain fluorescence? No? Yes? Modified Jablonski diagrams, right? Inter ISC and stuff like that. Yeah, all remember that? Interconversions and state like that? Good. Well done. Unfortunately, we are not going to be talking a lot about optical biosensors. We're going to this is just one slide to run about optical biosensors. Can anybody in this room tell me why is it hard, or why is electrochemical biosensing more useful when it comes to point of care and wearables? Okay. What's the biggest challenge? What is the number one desirable characteristic of an electrochemical biosensing device? Okay, fine. Forget about the specificity, forget about the performance. I gave you an electrochemical device. What is the most important characteristic that you need from it? Signaling, okay, yes.
Accuracy.
Accuracy, yes. Fine. Accuracy, specificity, sensitivity, that's all fine.
So cost efficient.
Cost, well done. Well done. Did this cost idea come from this like GIAN series?
Comfortable to wear.
Comfortable to wear. Can you give me a synonym for that?
Tolerability.
Tolerable, flexible. The word I'm looking for, it starts with a P.
Portable.
Yes, portability, right? So if you want something to be portable, you should be able to miniaturize the whole, right? So the problem with optical biosensors is it's not very easy to miniaturize to a dimension. Like for example, your electrochemical biosensors could be miniaturized to the extent that if you look at a CGM sensor, which I'll try to show you this afternoon, I will implant it in front of you. The CGM sensor, or in fact, during the break, I'll implant a CGM sensor and show you how the CGM sensor is implanted. A CGM sensor has got all the electronics at the back of it, right? Please don't ask me to look at my glucose readings because I've been eating very well here. So, but I'll definitely show you how the CGM sensors are implanted, right? So the problem is that you cannot miniaturize and have all the components in a very small real estate. You know, the term we use is real estate. You need a much more bigger real estate for the optical biosensors, and that makes it a bit more difficult to implement in terms of portability and on-site measurements and stuff like that. Although having said that, they're very sensitive. So you will find optical biosensors next time you go through the airport security checks. When they make you stand and do this and that and all different types of exercises, and they check your bag. They're basically using, they take a swab out of your bag and they look for things besides the X-ray thing. They also do the Raman measurements as well, and you would see that they take a swab and then they take it to a device there, and then they put the sample in there, and then they throw away that fabric, and then they use, so they have a Raman system sitting in there. So it's nice in terms of performance, optical systems, but portability is a big issue there. I mean, that's my understanding. Okay, transduction methods. Everybody happy with this? We covered all of this yesterday. I mean, I've got some few slides to show you exactly what it is, just to consolidate on amperometric measurements where you're measuring the current, potentiometric sensors where you're measuring the changes in potential, impedometric sensors where you're measuring changes in impedance, and voltammetric sensors again here. This is an example of slide to show you exactly how the signal response look like. So this is a typical voltammetry. You have cyclic voltammetry and square wave voltammetry. This is how a typical cyclic voltammetry signal looks like. As I said, you have this nice birdie-like structure. I mean, everybody who starts electrochemistry loves to see the first bird, a nice bird-like structure, because that gives a good indication about the health of your biosensing device. If you have your max and your oxidation and reduction peaks at the right place, your reference electrodes are working well, right? So this is how this is how a cyclic voltammogram looks like. This is a image showing you how a square wave voltammogram looks like, right? Again, you can see that these are all directly proportional, and that's how you normally get it for the analysis side of things. Amperometry, where you're measuring current, right? Measuring current over time. So this is here, this is your amperometry. This can vary. You can get steps, if the current varies, if the concentration varies, the current varies. So when we do continuous monitoring, you can have a profile which can last you over 24 days. I think that's what's normally done in a CGM device. And then you have got your electrochemical impedance here as well. As I mentioned, you have a sinusoidal AC potential signal here as well, which is good to probe your design architecture and everything like that, but it can also damage very thin films that you have. So you can then look into other methods such as differential pulse voltammetry and square wave voltammetry, which is given on your screen here. I won't go much into details. Everybody happy with it? Or does anybody want me to go over it one more time? Because I think we covered most of it. I'm just trying to consolidate it and just doing a quick recap of your electrochemical transduction methods. Everybody happy with it? Okay. All right, let's have a look at few of the electrochemical sensors, how these sensors actually look. And these are examples of some screen printed electrodes. If you had to look at how this would look like if it's placed in a beaker, this is what happens in a beaker. You have a working electrode, you have a reference electrode, and you have a counter electrode, right? And you're applying a current between the working electrode and the counter electrode, right? And you have a reference electrode which is there to keep the potential fixed, right? This is your reference electrode here. So how would you normally implement this? So talking about miniaturization, these are planar electrodes, right? These examples that you see here are planar electrodes, right? Again, you might be thinking, why is he teaching us so much about planar electrodes and biosensing technologies, although we want to learn more about wearables? All this technology that we develop here, right? When we are developing wearable devices, anybody who works on wearable devices will tell you they don't start directly with wearable devices. They start working and they optimize the protocols on planar substrates on materials that are much cheaper. And then this protocol is transferred to a wearable device. Therefore, it's very important that you understand the basic fundamentals, the instrumentation, everything that's involved in a point of care wearable, in a point of care biosensing device, so that you can then transfer it to a wearable device. And that's how I started my journey in wearable devices. I learned about biosensing devices first, working on planar electrodes and stuff like that, working on disc electrodes and stuff like that, and then I thought about, okay, how can I implement this now onto a wearable device? You will have differences there because suddenly your surface area will increase. Those are things that you need to take into factor, right? So gold may be suitable as a planar electrode, but it may not be suitable as a wearable electrode. But those are minor tweaks. You can change the different materials. If you see that between gold and platinum, your platinum electrodes are performing better with your screen printed electrodes, with your planar electrodes, then you transfer that onto your wearable electrodes. All right, so that's something that I wanted to bear in mind for those of you who are going to be venturing into this area of wearable technologies. The images that you see on your screen, these are basically 3 electrode setups. I mentioned you're going to have 3 electrodes which are most important. Where does the main action take place? Working electrode, counter electrode, and reference electrode, right? So this is how you have, and then you have an electrolyte bridge. This is basically a drop of solution that sits on top of this whole thing there. You can have, as you can see from the schematic here, you have your working electrode where the action takes place, your counter electrode, which is a semicircle. And as I said, as a rule of thumb, people normally try to have a much higher working, either the counter electrode should have a higher surface area or surface area comparable to the working, similar to the working electrode. Mostly the best we've seen the best case scenarios where you have much more higher. So if you look at platinum wires that are normally sold, they would have a lot of turns on them, a lot of knots. That's how they normally work. Okay, so that's the schematic, and this is then again how you bring that and how you implement this. So this is the first example of how we implemented our working electrodes, our planar electrodes straight to a microneedle array here. This is a schematic showing you that. So what we have here is basically you have the first layer, as I mentioned to you, we have SU8, which is out of which you make your microneedles, yeah, and we call them microneedles or microspikes in those days. We were calling them microspikes, I think. And then on top of that you have a layer of gold, right? So you have your SU8 and then you have your gold on top of it, and then on top of it you have your enzyme. Here, because you had gold, what we did here was we immobilized the enzymes using what sticks to gold? Thiols, right? You remember that example I gave you of SH groups? So we had a thiol group there. So we had thiol, we form a self-assembled layer of thiols, and then we do EDC and HS coupling. This is used to combine, this is used to get you amide linkages. So if you have a COOH group and you have a NH2 group, you can combine these two together with a condensation reaction where you end up getting, you lose a molecule of water and you get CONH linkages, and these are very strong covalent linkages there. So you have your self-assembled monolayer which are terminated with the carboxylic acid, and you have your enzyme which has got the amino groups. So your amino groups of the enzyme bind to the COH group of the thiolated compound, and then they are all self-assembled there nicely. And then on top of that, you put a epoxy polyurethane membrane. This was the first studies that were done, but these were done using, again, these were done in vitro, right? So what we had here is a microfluidic system. This was a paper that was published, and you can see from here that you have different concentrations. You keep on adding it because it's a microfluidic system, so you're doing continuous measurement. This is an example of chronoamperometry where you're measuring current over time. So when you start here, your currents drop down, both of them. There are two channels here, right? There are two different devices here. So you add then 2 millimolar, you get a step. Then you add 5 millimolar, you get another step. Then you add 10 millimolar, you get another step. 15 millimolar, 20 millimolar, 25 millimolars. So you can see there are two different devices. The reason you have different performance is because you're using a microfluidic system and you're pumping fluids at different flow rates, right? So you can see that this is such a much higher flow rate, and you can see here that these are the different steps you're getting here. All right, so this paper has been published, and this is also one of your suggested reading. This is just to show you what I just mentioned before, that once things work on the planar electrode, you can then try to transfer them onto to your wearable device. I call them microneedle here. Microneedle basically is a glorified subdermal wearable sensor, right? So you can easily transfer them, and you may have to do some tweaks, but it's something that can be transferred, and not only you can also publish them. And then finally, the other thing that we need to look, I think we are running a little bit of time over time, but I will be talking about performance metrics here. So I've already given you some examples yesterday. Do you remember the Clark grid errors, anybody? What did we use for the lactate sensors to look at the performance? Louder, please. Yes, somebody said BA. What does that BA stand for? Bland-Altman plots, right? Okay, good. So I'll just quickly go over this thing. So how do we look at the performance metrics? How do you look at the performance of your sensors here? Well, the important criteria or the important things that you normally need to report in your paper are sensitivity, limit of detection, selectivity, stroke specificity, response time and stability, calibration and reproducibility. These are the important metrics that you need to know and understand if you're going to be working on your biosensing devices, right? What are these? Sensitivity. I've shown you many of those calibration curves. You remember those response curves, calibration curves? What you do there is you have different concentrations. You measure the response or the current in response to these different concentrations, and then you do a plot. You do a fit here, right? And you see how far this particular slope is your slope of high sensitivity, right? And this is your here. This half of this is what you call as the limit of detection. When you have a low sensitivity, that's your limit of detection. This is where your blank signal is. This is where your blank signal starts. Your blank signal is never zero. Your blank signal will have a background current there. This is where the blank signal starts. So this is the dynamic range. This is your linear range. This whole thing, how far you can go, how far you can see the increase is what your dynamic range is. And after this point, it starts to saturate. You can see it starts to plateau there. Right? So these are how you calculate your sensitivity and your limit of detection. This slides, you will be getting a copy of these slides, so don't worry if you're not able to get these slides, but this is something that you need to characterize for your device. I'm not here and I'm not expecting all of you to absorb everything, because I know I speak really fast and it's virtually inhuman for me to expect you to absorb everything. But please do have a look at the slides after you finish, when you have some time in the evening or whenever you look at the lectures, because this is important piece of information that you need to know about your devices, the performance metrics of your devices. What is the lower limit of quantification? What is the higher limit of quantification? What is the limit of detection? What is the sensitivity? What's the linear range? What's the dynamic range? At what concentrations do they plateau? When does it start to saturate? You need to know all of these about your sensors before you can bring them into the real world, before you can do any translational research. You need to optimize, and even with in vitro studies, those of you who work on in vitro, stop working on PBS. Identify what your final sample matrix is going to be. Are you developing wearable sensors? Okay, so you're going to be working on skin interstitial fluid or sweat. Why don't you use artificial sweat? Why don't you use artificial ISF? You have protocols for all of these available in the literature. If you can buy blood as a reagent, if you've done the training and everything, if you got the research ethics approvals, use blood, use whole blood, use plasma, use serum, and then develop it. Because what happens is, if you develop everything in PBS, phosphate, in 7.4 pH and things like that, you're basically once you start working in real samples, you will have to go back to the drawing board and redesign your sensors, right? So very, very important to do all your studies right from the start in something that is very similar to the final application area that you're going to be working in. Okay, talking about selectivity and specificity, good examples here. Selectivity is more when you have single analytes, how specifically it binds to it. Selectivity is how selective it is, how good selectivity is. Normally when you have sensors, for variable sensors, you want something which is it shows you good selectivity but not very good specificity. Why? Because you wanted it to bind and then you want it to detach so that you have those sites being regenerated, right? So this is a slide, and there's a paper here which explains selectivity and specificity very nicely, which can also be seen from this particular diagram here. I've given you the example of antibodies here, and these are more like molecular imprinted polymers, right, to give you examples of sensitivity and selectivity that you have on your screen. And then response time, how long does it take to respond to the change in concentration? That's a very important parameter. You don't want something where the response time is, it takes about 15 minutes to get the actual value, right? Okay, the incubation step may take 30 minutes, but once the incubation is done, how long does it take you to get the sensor? Because think about it, you're developing devices. If your devices are going to take 4 hours, 5 hours, then it kills the whole purpose of using devices at point of care sites, right? So you need to have devices which have got a very short response time. Then the other thing that you need is stability, performance of the device. Now, how does the device perform? Has it got regular, you measure it over a certain number of days and you see how the performance is over days. Is it a stable performance or is it a unstable performance? One day it's giving you high value, one day it's giving you low value. You normally measure the baseline. So this one is an example of unstable one, and this one is an example of how the values are very stable. There might be slight variations, there might be slight degradation, which you can account for. If there is a trend, you can bring in the machine learning and the AI tools. But if there is no trend there, nobody can save you there. Does that make sense? Okay, and then finally looking at the calibration. I mean, we've been through many of these. I've shown you, I don't know how many of these calibration curves. This is how you get your calibration curves. Look at this nice calibration curve that you have here. Look at these ones here, right? What are these calibrations? So the one thing is calibration, right? This is where which is more like a dose response curve, right? The other one is here. This is an example of, this is the kind of data you would expect in a journal, right? To get something without error bars, if you cannot publish, if you don't have error bars in your data, you need to have n equals 3 and you need to have multiple points and you need to have error bars. If you don't have error bars in your data, you cannot publish your data. Simple as that, right? Any journal, any paper that comes to me for review which doesn't have an error bar, I know that is made up data, because in real world you always get some variation. Even if it's a minor variation, you get some variation there. So this is the example of error bars shown nicely in the data here. And then talking about reproducibility, I mean, look at this. Day 1, measurements done over day 1, day 3, day 7 and day 14, right? This is clearly an example of a continuous glucose monitoring sensor here. I mean, there's slight variation there, but by and large, look, it's very, 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 very reproducible, right? That's nicely shown in this particular data, and this again is nicely described in this particular paper for which I've given the DOI in the slide here. All right, I think that brings us to probably the last or second last slide, which is the Clark grid error. You remember this Clark grid error? Everybody remembers CGE, Clark error grid or Clark grid error, right? This is basically, as I said, when you're looking at your measured blood glucose with your biosensor and looking against a reference method, which is a gold standard, if ideally all the values should fall on this straight line. The nice thing with glucose sensing is there's a little bit of a room to maneuver, right? And this room for maneuvering actually gets narrower as we move from higher concentrations to lower concentrations, because as you move towards lower concentration, there is less room there's less margin for error, right? And as you can see, because we do not want a case of hypoglycemia, we don't want our sensors to be overreading when the readings are down, right? And setting this. So this is the Clark grid error, which is mainly used for glucose sensors. And then finally, bland Altman plots, right? We covered this as well yesterday. Ideally, all the points should fall on this straight line here. This is again looking at the difference between lactate and the average lactate here. Ideally, everything should fall along the straight line here, and these are your confidence limits. You have 95% confidence limits. As long as the points of majority of the points are falling between these two blue lines, everything is fine. These ones are the outliers which need to be, I mean, in this case there's only 2 outliers or maybe 3 outliers which need to be investigated, but you can call them outliers and just leave them alone. But if you have many points which are outside, beyond, if you have more points outside the blue lines, then that's a worry. Then your sensors are not the best, right? So you need to think about that as well. And these Bland-Altman plots are normally used for looking at other types of sensors other than glucose, lactate, ketone sensors, and all the other things are normally done using Bland-Altman plots. You also have something called as violin plots that I'll show you when I show you the p-tau results. So kind of now almost towards the end of the talk here. So when you move from the lab to the clinic, there are certain factors that you need to account for. Biofouling is a big problem. And as I said, if you understand the trend of biofouling, you can bring in your machine learning and AI tools and you can use that. I showed you yesterday a case study where we used AI tools and we could make much better sense of the data. Miniaturization. How do you miniaturize? I mean, I showed you I haven't done that, but I have one lecture just dedicated for animal studies to show you the challenges there, because nobody has done animal studies. Nobody has used wearable sensors on animals, right? And the animals actually deserve more to have wearable devices because they cannot give you a visual assessment, feedback, and you cannot have capillary blood from them. So there's a lot there that needs to be done in terms of miniaturization. And then, as I mentioned, you need to have the right approvals. And how do you integrate them into these apps? I mean, this is the latest fashion these days. So this is my last slide. What we've done today is we've recapped on biosensor types. I hope I don't have to repeat it again. I hope I've helped you consolidate all your learning that you've been doing over the last 4 lectures. We've seen the values, the importance of translational value and the challenges that you have when you translate a device from the lab to the clinic. And if you have any questions, I'm more than happy to take them. I'm sorry for running over time.
It's okay. Any questions?
Actually, sir, in the molecular imprinted polymers,
Yeah.
you have mentioned we don't require washing steps.
No.
So, sir, how we can regenerate the sites there to bind another antigen? How we can reuse those devices?
Excellent question. These are point of care devices. When I say point of care devices, we are looking at disposable devices, number 1. The other thing you need to understand, clinical samples, you never reuse the devices.
Okay.
I would never reuse a PCB electrode, no matter however good cleaning protocol I have, if I'm using it with human samples, because there's every chance of having cross contamination. So you don't need to regenerate it. You can just dispose it off.
Okay. Thank you.
Yes, but this question, the same question with wearables would be very, very interesting. So in that case, you can have wearable devices because at certain point they will get saturated. Then you can change the device and use a new device, or choose molecular imprinted polymers that can bind and unbind. So as I mentioned, between selectivity and specificity, that are specific but less that are selective, so they go and bind, but then they unbind later on, so that you can free the site, and then you can have more. You can do more continuous measurements. But ideally, you would like to have something that works over 6 hours or 12 hours, and then you dispose them off.
So for removing antigens, we require again the some kind of proteins and enzymes, right?
For removing what?
The analytes which got binded into the sites. So to remove them, we require enzymes and proteins.
No, we don't need it.
So how can we remove them?
No, for wearables, you've used it. It will give you a value, and then you dispose off and get a new one, a fresh one. Yeah. Good. These are really good questions. It shows that you're thinking about it. Any more questions? No? Everybody's eager for a cup of coffee or tea?
Yes, any more questions? So I might have a few. Actually, so this molecular imprinted polymers, they seem like a very good method of sensing. So is there any limit to it? Like you have tested it with vancomycin and any
We've
other-
tested it with
so what-
vancomycin.
and some protein.
Yes.
Yeah. So what I mean, can it detect any analyte in the world or
In
target?
principle, yes. With
In
the right
principle.
choice of molecular imprinted polymer.
Okay.
In principle, yes. Because
Okay.
there's a wide range of polymers that are available.
Okay.
It depends on your target analyte, how
Size of the
hydrophilic,
target.
hydrophobic it is.
Okay.
So you have to be very careful. That's why I stressed a lot on the interaction mechanism. And I try to explain it by you explaining the different forces that act, the hydrophobic forces, the electrostatic forces of attraction, the hydrogen bonding. So you have to actually, it will be nice to have this whole thing modeled nicely,
Yes, yeah.
to have a model which can help you then choose your polymer accordingly.
Okay. Okay. So first you model it and then check out.
Yeah, you
Then
model
you-
it and you model it with, so you will have polymers which are, this polymer is suitable for hydrophobic molecules, this polymer is suitable for hydrophilic molecule. The thing is with electrochemistry, you have to have it it has to be very aqueous based.
Yes. Yeah.
That's where the challenge is. That's where you bring in zwitterions and all those things.
So what, like, for the benefit of the participants, what sort of tools, modeling tools do you generally use for this? Just if you
I
can
mean,
name
you
a few.
can just use the Gen AI tools that are available. You can use anything as simple as ChatGPT and DeepSeek and Gemini if you want to just understand it first of all, and then you can use PyMol.
Okay.
And the reason I say that, it will save you a lot of time to model it, to understand it through the generative
Traditional,
AI
yeah.
tools, and then it will suggest you how to model it using the PyMol and stuff like that. That saves you a lot of time, because I know that this can be very time consuming when you sit down there and try to model and then you try to identify the sites. It's very hard.
Yeah.
Why not use the tools that are available at your hand, at your disposal?
Okay. So these violin plots you will be showing later on, I guess.
I'll be showing you tomorrow.
Okay. So in this lecture, just like as sir has said, we covered different biosensor types, especially the bioreceptor sensing using MIPs, molecular implanted polymers. And then we also looked at a very important aspect is if you develop a new biosensor, then you have to look at all the plots, Clark grid error, blind Altman, and all the characterization. These are the typical characterization steps, calibration that you need to follow for your own biosensor to show and to understand that whether it is working successfully or not. So with that, we end this session. There is tea outside, so let's reconvene again after a short pre-break, approximately around 11:45 or 11:30. Thank you. स्ट्रीमिंग ऑफ कर दिया ना रिकॉर्डिंग ऑफ
I'm ready when you're ready. Start. Okay. All right, everybody, welcome back. So this particular lecture is going to be mainly on epidermal variables, right? We're going to go back to this area of epidermal variables. As I said, not particularly my area of research, but I'll do my best to explain what the fundamentals behind the epidermal variables. We're mainly going to be looking at sweat-based epidermal variables, the platforms, the materials that are used, the instrumentations that you require, and the applications in this particular series we have here. Okay, so in terms of course structure, this was actually meant to be for tomorrow. That's where you saw the different date on the main screen. We've just brought it forward because I want to keep that dual receptor thing, which involves combining bioreceptors lecture for tomorrow morning. So I've just swapped the order around a little bit, but it's still going to be considered as lecture 5. Okay. So the learning objectives for this particular lecture are mainly to understand what the composition of sweat looks like, what's its relevance. We've already touched on a little bit. We had a bit of an outline when we discussed about epidermal variable sensors on day 1. We'll explore the different sensing platforms that we have. What are the different types of sensing platforms for sweat-based epidermal devices? What materials could be used for building those devices? And we're going to be looking at how signal conditioning and power strategies can help us improve the performance of the sensors, and review some of the clinical and field applications that have been performed with these epidermal variable sensors. Okay, first of all, let's just do a quick recap as to why sweat is an important biofluid. Why is it important? What are the advantages of sweat as a biofluid? Well, the first most important thing is it's non-invasive, right? I mean, you don't need to you don't need any sampling device to collect sweat, in the sense that you sweat naturally, right? The problem is when you can't sweat, right? So I think most of the people who work on sweat-based sensors, they also kind of employ methods in which sweat can be generated. But again, as I said, the advantage of sweat as a biofluid is the most important thing is it can be accessed in a non-invasive manner, and it's readily available as well. What does it have? It contains most of the metabolites because it's all coming through your sweat glands. It contains glucose, it contains lactate, it contains cortisol, it contains electrolytes. Easily accessible, has these analytes. Is that all that we need? One more thing we need is there should be a good correlation with blood. That is a big question mark here. It correlates with blood for select analytes. It does not correlate for all the analytes. That's the problem we have with sweat as biofluid. I think that's why sweat-based glucose monitoring is not very common, because there's not a good correlation of the concentration of glucose in sweat when you compare that to that in venous blood, right? While in skin interstitial fluid, you have a much better correlation when you compare the concentration of glucose in skin ISF versus venous blood. So the challenges that we have when we talk about epidermal microfluidics or epidermal variable devices is, number one, the sweat rate will vary. And the other thing you can have is you can have contamination with something that's already on the surface, right? A lot of you use deodorants and stuff like that. Imagine you try to collect sweat and then you're spraying your own deodorant, and you have a sensor which is meant to be monitoring alcohol. Yeah, you've already contaminated your surface there, making it really hard, right? So this is thing. So if you look at the composition of sweat on your screen, I'm sorry for the small fonts, it's mainly made up of water. It has some sodium, some potassium, and also got some urea there. And I think I showed you some examples wherein the variable epidermal sensors were used for monitoring urea in sweat as well. Okay, sweat as a biofluid, there's been a lot of trend towards measuring urea in sweat, and there have been the different methods that have been used for this. People have used amperometric methods, people have used impedimetric methods, potentiometric methods, voltammetric methods, piezoelectrometric methods, Raman spectroscopy, which is again an optical method, fluorescence spectroscopy, electrochemical luminescence, and colorimetry. So different methods here, and you can see most of this has been done In fact, this one is your electrochemical. So most of it is done using electrochemical methods. You have this mass-based, which is your piezoelectrometric method, and the optical here is just this little quadrant that you see here. Okay. So again, it brings us back to the same question: why are electrochemical sensors more popular even when it comes to epidermal wearable devices? And the answer to that is portability, ease of miniaturization, right? That explains this large fraction of this large pie of this whole thing being more electrochemical sensor based compared to the optical here and the mass based. Piezo is really hard. This has actually been pulled out of a paper which is on sweat analysis for urea sensing, which is in a paper published in 2023 in Analytical Methods. This paper should also be made available to you through the folder that will be made available by Professor Khalid, or it's already been made available. Okay. Who are the pioneers who work in the area of sweat? I think we've got 3 main people who work here. One of them is a very young guy, Wei Gao. Some of you idolize him, I know that. A young guy, he's just started doing it, working in this area of, he's from Caltech in Pasadena in the United States. Actually, all the 3 of them are from the United States. You know, you may ask the question, why are all these people from the United States and why do we not have anybody from Asia or from Europe? And the answer to that is there's a lot of funding that's available for adventurous research which are kind of, you may have, which are high risk research projects, and those are normally funded by US funding agencies more than you would find in Europe or in India. In Europe and India, they like to have projects which have got minimum risks, something that's been tried and tested, something where you have some proof of concept studies or valid data there, and you don't get anything to do kind of anything under the blue sky kind of research there in Europe and in Asia as well. The main big player in this area is Professor Joe Wang. Joe Wang is the book that I recommended in the last lecture was that by on analytical methods or analytical electrochemical methods was by Joe Wang. He's the head of the Center for Wearable Sensors. He's a distinguished professor of nanoengineering in the University of California in San Diego. He's also a director of the Center of Wearable Sensors, co-director of Center of Mobile Health Solutions or Systems, fellow of the Royal Society of Chemistry, fellow of the Electrochemical Society, and he works in this Department of Chemical and Nanoengineering in the University of California in San Diego. A fantastic person. I've heard some of his talks at Imperial and at various World Congress on Biosensing conference platforms as well. Another person who works in this area of epidermal wearable devices is Professor John Rogers. John Rogers is based in the Northwestern University in the Department of Material Science and Engineering in the United States. And again, his area or his expertise is mainly on soft electronic and microfluidic systems for skin. So he works in this area, which is popularly known as epidermal microfluidics, right? This is where you try to address the problem of variability in sweat. So rather than just monitoring sweat in real time, you collect them using a microfluidic system and then you analyze them, right? You don't have to actually, when you talk about wearable devices, you don't have to measure every analyte in real time every second, right? Even the best case example that you have, that is the commercially available continuous glucose monitoring sensors, they measure glucose and give you values. They measure glucose, I mean, they have the capability of measuring every second or doing multiple measurements every second. But what they normally do is they measure and give you an output value. That value changes every 15 minutes. Okay, so these are the pioneers that you have here who are working in this area. So if anybody wants to work in these areas of epidermal wearables, or even for that matter subdermal wearables. In fact, I know Professor Joe Wang has also published some papers on microneedle based subdermal wearable sensors as well. So it's to keep on looking at which biological matrix for what would be more beneficial, and that's how they make a call as to what to work on. This is Wei Gao, a very young chap here. I mean, you can see his very flexible kind of sensors here. You can see it's on a flexible material. You can see this those lines there. Those are the basically showing you and these are some adhesives to stick onto the skin surface, and you have these, this is your working electrode, this is your, outside this is your counter, and then you have your reference electrodes as well, and I think they've got multiple working electrodes in this particular setup here. So the different sensing platforms that you have for sweat are tattoo-based sensors. People have made tattoos, made up of gold nanoparticles of graphene, PEDOT-PSS, which is basically, this is polystyrene sulfonate, and the other one is a thiophene here, polyethylene dioxalate thiophene. And then people have also used microfluidic patches. As I mentioned, I'll show you some of those microfluidic patches, very thin microfluidic patches, which basically have got reservoirs to collect the sweat and then to do the analysis. Another one which is very popular in India, there are many people in India who are working in this area, is paper-based microfluidic systems. One of my previous colleagues, Amit Asthana, who's in Hyderabad, is working a lot on paper-based devices. And I know that Bill and Melinda Gates Foundation have funded a lot of projects in India which is based on basically paper-based microfluidic devices, or paper-based devices for point of care and wearable devices. The pioneer for this, of course, was George Whitesides from the US again, and I think he's got some projects running wherein the projects have been funded by Bill Gates and Melinda Gates Foundation. We're also going to be looking at different platforms, bandage style flexible electronics, where you have like a bandage and that's got flexible electronics. Has anybody heard about dissolvable electronics? So the problem with electronics is that electronics, they generate a lot of waste. I mean, you might have seen from the pandemic where everybody was having those laminar flow devices, and some of them were having electronic units. They had the PCBs which had got you could easily see the capacitors and then the little screens and stuff like that. Even the people who use pregnancy tests at home, or people who use glucomonitors, blood glucomonitors, there you just have the strips that are being generated. The rest of the electronics are being reused, but there are a lot of laminar flow devices which come with electronics, and the biggest problem there is the electronic waste that you have. That's why point of care devices are kind of going, people are looking for alternate ways, especially methods which are in accordance to green chemistry. You can look at dissolvable circuits, dissolvable electrical components. People are looking at natural material based devices, silk, paper. PDMS is not natural. PDMS is hard to dispose of. It's an elastomer. So there are these different materials that are being used for these bandage style flexible electronics as well. And then there are these fingertip passive sweat sensors, wherein you take the sweat and you transfer it to a finger. I showed an example of this yesterday. For some funny reason, I also have seen this kind of thing wherein you have the microneedle and sweat based hybrid systems, something which is measuring under the skin and on top of the skin as well. So this is like more like a hybrid system here. So the microneedle based sweat hybrid system that you have here. Okay, let's start looking at some of the examples here, and then we'll have 4 specific examples, but let's have a look at this one here, which is what we call as an OECT, which is an organic electrochemical transistor device. Yesterday, the transistor device was explained by Professor Khalid. He mentioned about having a gate, a source, and a drain, right? And the gate part is the part where your solution normally, you have an extended gate, that's where your solution normally sits, and then you have a source and a drain, and you normally apply the potential, or you either monitor changes in the potential, or you keep the conducting channel, the conducting current, the conductance fixed, and you measure the changes in the potential there. So it's a potentiometric method for monitoring stuff. So that's a transistor here, and the other rest OEC stands for organic electrochemical transistor here. So this is basically a highly sensitive biosensing device that has been produced based on PEDOT-PSS, which I mentioned is nothing but a thiophylline and polystyrene sulfonate based organic electrochemical transistor. You can see that the limit of detection, you're getting it is in micromoles, which is fantastic. How you make these devices, again, you're having a flexible material here because you're talking about your sweat side of things. You need to create channels so that you can collect the sweat. So they're using laser induced micro patterning. So you can draw the lines with laser, you can actually ablate, and you can create channels. So they use this labor induced micro patterning to get these kind of channels on the PEDOT structures there. And then basically this is a single use epidermal sensor here. You can see there's a reservoir here, and on the back of it is your PEDOT layer, which is basically an organic electrochemically active layer, and this was published in the ACS sensors there, and you're getting good limits of detection here, which is in the micromolar range in this particular case here, right? So this is a nice paper which was published in ACS Applied Materials in 2024. It's one of the recent papers that have just come out. All right, and then I'm going to talk a little bit about epidermal microfluidics. You can see the man himself, John Rogers, with some of his device, especially in the form of tattoos that have been stuck on his face. We've got some nice little tattoos that he's got there, and he's also holding a PDMS device there. And this is a nice little device which is sitting on your forearm. What it does is it's basically again got a series of, so it's got different multiple layers. So you have the sensing layer which should be on the top, and you have the bottom layer is more like a microfluidic layer for collecting the sweat, and you collect the sweat, and then you have 4 different reservoirs here. You can see the different colors there, and they're measuring things in a colorimetric manner. That's a nice thing about epidermal microfluidics. Now colorimetry clearly is not an electrochemical method, it's more like a visual method, it's more like an optical method there where you see a change in color. Very, very interesting, and I can give you examples. When we had this whole incidence of animal influenza, birds were being culled in the UK, and so we were asked to design projects which would allow us to ensure that the birds are not culled or killed just under thinking that, oh, you don't need to wipe off the entire flock that you had on a farm there. So people wanted to have visual, for farmers, what's the easiest way to develop test for farmers? Something that's visual, right? A color change. So we had these devices which were based on APTA sensors, and they were more for visual ones. So what you would do is take a swab of the backside of the bird and then dip it into an Eppendorf tube, and then which would have a solution, and then you would see a color change. So that's how you would transfer the biomarkers from the backside, which is basically of the bird, and transfer it into the Eppendorf tubes, and then you could see the color change. That's a nice way of doing colorimetry. Is a nice way of it's a simple way. It gives you qualitative information. It tells you yes or no, but it doesn't tell you about the quantitative information. It doesn't tell you how much, right? So this is a nice colorimetry thing which tells you how much basically of this hydration is there, how much of urea and stuff like that are there, but it doesn't tell you, it tells you if it's there or not, but it doesn't tell you about how much of it is there. And then this is basically another epidermal microfluidic device which also has a pump. This is so you have the pumps which are designed on top of the it's like more your pressure pump. You just apply a thumb pressure and you push the device so that it gets distributed into these channels that you have here. So you have your sweat that's collected here. You apply the pump which is designed on top of this polymeric layer. It's the pump inlet and pump outlet. You apply pressure, and it then pushes everything inwards towards these radial channels here, and you can see what's happening here is it starts to fill up all the different channels, and you have the biosensors underneath the channels which can do you can do multi-analyte sensing as well. So this is a nice interesting paper, again published in one of the journals that we all dream in publishing, Nature Communications. This was published back in 2019, again by John Rogers. This is an example of where iontophoresis is being used. So we spoke about the gluco watch yesterday. So basically what you do is you apply a current. When you apply a current, then it kind of extracts it makes the ionic flow in the direction so that it comes out of the skin, and then you induce sweat there, and then that's how you normally measure this in using the ionophoric measure through iontophoresis and measuring the current. You can get your epidermal variables as well. So the gluco watch falls into the category of the gluco watch thing that you said, and this is some work that has been pioneered by Patricia Pat Connolly from Strathclyde University. If you want to look into this work, you can easily follow her up on that particular website there. It's nice. It's very nice in the sense that, again, you don't need to extract anything. You don't need to push anything under the skin. There's no needles there. It's a very neat way of conducting things. But again, limitation. There's a limited number of analytes that can be measured using epidermal variables. So what are the materials that have been used for your epidermal sensors? Well, the people have used things like PET, PI, which is polyamide, PDMS, EcoFlex. These are all very flexible materials. You can actually make sheets. You can make rolls. If you can make rolls of anything and then cut them down into something that can be easily wrapped around your finger or wrapped around your wrist or wrapped around your arm, makes life much more easier. Anything that can be made in rolls is a scalable technology. You can make multiple rolls, and it's low cost as well, right? So epidermal one of the good things with epidermal wearable devices and the technologies that are used for making these epidermal devices are those technologies are scalable devices, because you use substrates that can easily be kind of formed into rolls. And then on top of that, you print your layer or your conducting material, and normally it's not as if you're using real gold. It's just 50 nanometers, 100 nanometers of layers of gold, silver, carbon nanotubes, or graphene that can be printed on top of this rolling material. And it's, you can imagine, and in some cases what they use is just a glorified inkjet printer. So you take your inkjet cartridges, replace them with cartridges which can give you gold ink. You can get gold in the form of ink. You can get silver in the form of ink. You can get carbon nanotubes and graphene in the form of inks. So you can replace the cartridges with those ink cartridges made up of gold, silver, carbon nanotubes, and graphene, and you can print your own devices. So you can literally, if you get the roll, if you order the roll on Amazon or this thing, and if you have a printer cartridge which you can modify, you can make them in your own home settings. Okay, and then you can use these to then, and then you can, on top of that, if you're trying to monitor ions, then you need an ion selective membrane. If you're doing enzymes, then you can also get enzymatic inks as well. You can mix the enzymatic inks in your conducting inks, and you can have something wherein they are nicely, you can form thin layers there. And you also have this whole option of printed electronics. This is what I just mentioned about. You can have screen printed electrodes, you can have inkjet printer based electrodes, and you can also have aerosol jets wherein you can just spray them and then dry them. Somebody was talking about electrospinning as well. You can also use electrospinning to electrospin on top of your substrate material. This is another example that we have on wearable sensors which has been made of different materials. This probably work is come from, again, Baig, right? This is basically published in Nature. Sometimes it's a mystery how a person can publish so many papers all in Nature. That just shows the quality of the work. It's Nature around these. So people who know the field of publishing, you have journals which have got very high impact factor journal. So all the Nature journals, the science journals, impact factor is good, but it's not the only thing, right? But for somebody to publish continuously in journals, especially Nature thing, some of it is reputation, and most of it is the good quality of work that comes with it as well. Okay, so this is where he's basically shown how he's used a fully integrated wearable sensing platform using a PET device and substrate. Sorry, this picture basically is to show you wearable sweat sensors made of different materials. This one has come from the group of Baig. The second one is where you have embroidered. So you can use fibers, with conducting fibers. You can use a sewing machine, and you can have a sewing machine and you can make your own electrodes on fibers as well. That is a nice and neat way of making wearable devices. This is, I think, probably the work of done by Firat Guder from Imperial College again. Again, very, very neat. You can see here he's printed the counter electrode, the working electrode, and there's a reference electrode here which is not very, very clean, nearly visible. And this has been printed on a nice cotton t-shirt as well, right? And then these are some examples of sensor patches for monitoring sweat. What they've done is it's basically a band-aid pad on which you have your sensor which has been printed there outside and inside. You can see here, this is the inside one. Somebody was talking about wound healing. So if you want to monitor wound healing, this is a nice way of having sensors which can have iridium oxide sensors to look at the low pH or the pH dropping down, and they can also have sensors which can measure glucose as well. I think somebody is looking for wound healing. Yeah, that's wound healing. So you can, this kind of a thing will be very useful here. The references are here on this slide here, so you can pull out these references once you get the slides. Yeah, so it shouldn't be a problem here. And then you have this example of this sweat analysis thing, which is based on a foldable all paper substrate. Again, this is an example where paper has been used as a substrate, and you have this sweat analysis design there. And then coming to this example of silk, I mentioned of silk to a few of you yesterday. Using silk based carbon materials. This is your silk based sensors which have been used as a wearable electrochemical sensors as a working electrode. That's the example of the silk one. And the last one is basically a wearable lasers. This is a laser engraved sensor which is again used for sweat detection for different analytes. I think the most common one is urea. I think urea is used a lot for sweat detection because it's got its own challenges, and I think there's a good correlation of urea in sweat and urea in blood as well. Transduction mechanisms similar to what we saw this morning and yesterday. As I said, the basic principles that we've seen, Potentiometric methods are used for ion sensitive electrodes. Of course, as the name indicates, ions, meaning that you're looking at sodium and potassium ions, right? For these, you normally have those little membrane like structures which are very selective to the different ions. They're more like size exclusion membranes. They are sensitive to the differences in size, and they are able to differentiate between sodium ions and potassium ions based on just the size, right? So those are membranes that are used in ion selective electrodes. And then amperometric sensors. I mean, I think you must be bored now with listening to glucose sensors, but that's how it works. Glucose sensors and lactate sensors, which are based on enzymes. The same transduction mechanisms are used in epidermal wearable devices. In terms of impedance, you can measure hydration. Most of you might be having a weighing scale at home which measures bioimpedance. Anybody come across that? So if you can buy these off Amazon these days. You have a weighing scale which will also tell you about your BMI. How does it do that? It can calculate the total water in your body and calculate your weight, calculate your dry weight, and it can calculate your muscle weight, calculate your bone weight. So it does some impedance measurements. It's called bioimpedance measurements. You step on the weighing scale, and it does a lot of calculations, and then it tells you, okay, your BMI is this much based on your weight, your height, and the dry weight in your body, your muscle weight and stuff like that, and gives you your BMI, right? Has anybody tried that? Has anybody used that in home settings? No? You're all very young. Wait till you get to past your 40s. I think everybody will start using them. Colorimetry. Colorimetry is very, very useful. I've already given you examples wherein it's a nice way of visual detection, point of care. If you see a color change, it should be a significant color change, ideally from blue to red, green to red, and things like that are very much visual, although it might be an impairment for people who are suffering from color blindness. Okay, the instrumentation for this will be introduced by Professor Khalid. Just to let you know what are the different kinds of instrumentations that these people used, because you might have seen some of those pictures where they have those very thin layers on which they have the different forms, and I showed you some instrumentation when I introduced you to the slide on biosensors. You have some instrumentation at the front end which will involve things like filters, amplifiers, and ADC units, analog digital converting units. And then you will also have some microcontrollers. You're getting data coming out of it. You don't want the person to be collecting the data and then processing the data. So you need to have a microcontroller which can do all the processing for you, and the processing unit can then smooth the data. You can have your machine learning and AI codes on it, which can then help in making more sense of the data, and it can predict a value. Yeah, you can get a predicted value based on all the microcontrollers. So you can have data handling there, and then you can have all of this BLE and NFC, which Khalid will be able to explain you much better when he has his lecture on the instrumentation side of things. In terms of powers, you normally could have a battery there. It could be a tiny little lithium battery there, or you could have, we have also shown you examples wherein people have started integrating biofuel cells, lactate based biofuel cells. You can also integrate that, and you can make these into a fully integrated sensors there. And then how do you collect the data, right? The data will be normally accessed through the cloud. You can have a smartphone, or you can have cloud based analytics. You can have what we call as an iCloud, wherein you have everything goes into the iCloud, and from the iCloud it can be relayed onto your smartphone using a Wi-Fi signal or things like that. So these are the standard parts of a wearable sensor that you need to this thing. Main focus of this whole GIAN series is to kind of make you aware of these different components that go in to build a wearable sensor. And then looking into applications in health and fitness, they have been used for different forms of applications in health and fitness, right? I mean, most of these kind of sensors, wearable sensors, are used by fitness freaks, or I mean, now these days there's a big I would say the people are becoming more and more alert. People are using sensors for very different reasons. People who are, I mean, it's all started off by trying to address a problem. The problem was diabetes. It all started off by addressing a problem of type 1 diabetes, right? When you try to look into type 1 diabetes, the problem there is it's more in juveniles, more in kids. So you needed a carer there. So you needed something that could give you continuous reading, and then something that could give you could take the control of you, and then decide for you if you needed an insulin shot or not, if you needed to adjust insulin dosage or not. That was meant for type 1 diabetes, right? That's how the whole concept of artificial pancreas came in. That's something that, again, Professor Khalid will be talking about, right? So you can have that, that that was the main application, and then it moved into type 2 diabetes. People want to see how the profile looks like over the days, right? Type 2 diabetes is more lifestyle diabetes. This is something for which we ourselves are responsible, right? You can have insulin resistance, you can have, you can basically have diabetes because of very sedentary lifestyles. I mean, I've been sitting on this chair for the last 3 hours, not getting up at all, right? And I would have preferred doing this lecture walking around. So I know if I measure my glucose now, the numbers wouldn't look very good, right? So type 2 diabetes is more lifestyle related diabetes, more sedentary diabetes there. So now the whole application of these, especially for these wearables, is more towards well-being, and well-being includes measuring your metabolites like glucose and lactate. People are running around and they're checking the lactate levels. People are checking their glucose levels. People are checking to see how long are the glucose levels staying within the normal zone, and how long are they staying outside the zone. So people are becoming much more aware of this health problem and making sure that they're looking after their own health care. So these wearable devices are actually helping you decentralize healthcare from big hospital settings to a GP practice, now in your more home care settings there. Okay, and as I said, so these wearable devices are finding adjunctive glucose monitoring for diabetes, cortisol for stress and circadian rhythms, and as I said, urea, one of the biggest biomarkers that are normally explored by the epidermal wearable sensors, which is a bioindicator or a biomarker of chronic kidney function disorder. That's where you measure urea there as well. What I'm going to do in the next part is run you past 4 different papers that have been published. One was published in 2018 by Hong et al., which is basically on using epidermal devices for looking at glucose and the vital signs patches. And then Sempionato, I think this is Joseph Wang's group, which published in 2021, fingertip sweat sensor. And then Mishra published the kidney biomarker, again, which is a glorified name for calling urea biosensor. And then we had Ian et al., which is an example of how lactate was monitored, and they also used lactate was monitored. Lactate powered biofuel cells were employed for this particular study here. Okay, so this is the first paper. This is by a group from, I believe, which is from South Korea. Looking at the names, Hong was the first author here, and they developed this biosensor, which is on multifunctional wearable systems that are integrating sweat-based sensing and vital sign monitoring to estimate pre and post exercise glucose levels. So clear-cut, simple thing wherein you have a wearable device for monitoring glucose before the exercise and after the exercise, just to look at the device. So you're having a disposable device here. You use it, once you've used it, you can then dispose it off as well. But it's good to, again, this is continuous monitoring, but this is continuous monitoring over a certain duration. It's not like 14 days continuous monitoring. It's monitoring over a certain period of time which is pre-exercise and post-exercise. That's it. Just monitoring those peaks, just to see how much does your sugar level actually spike, right? That's your case study. That's the first case study, which is done by Hong et al. And this paper is available for you through the organizers. And then there's a second paper which was published in 2021 in Nature Biomedical Engineering, another journal that all of us aspire to publish into. This is by Joe Wang's group, and this is about an epidermal patch which is used for simultaneous monitoring of hemodynamic and metabolic biomarkers. A very nice way of saying, when I say metabolic biomarkers, I'm mainly talking about glucose, lactate, again urea, ketones, and sometimes they have also used for monitoring alcohol as well. So you can see they're looking at consumption of glucose via glycolysis, and they're looking at oxygen depletion and lactate generation as well. This is a good continuous and simultaneous acoustic and electrochemical sensing. So they're also doing using the acoustic sensors there as well. Those are more physical sensors, right? Surface acoustic waves. They're used more for acoustic sensing, and there are integrated wearable devices which should enrich and understand the body's response to daily activities, again for prediction of abnormal physiological changes, right? As we age, we want to look at how the physiology is progressing. Our metabolism slows down, so it's nice to have devices that can give us an indication about our metabolism. This then helps us to make changes in our in our lifestyles as well. You know, more exercise, less food, and things like that. This is all possible using epidermal microfluidic patches, epidermal wearable devices. This one is an example which I've already shown. I mean, I like this a lot. This is by Mishra et al. And this was published in ACS Census. This is again another good journal. It's got an impact factor of more than 10. It's a nice journal, and it's relatively new compared to the other ones. And this is where they've used colorimetry here. You can see you've got the circle here, and then you've got those 4 different patches. So you're actually getting n equals 4 readings, and you've got 4 different dots you can see which are arranged radially. And this is your sample collection point, and then this is how you activate the pump. So you've got the 4 pumps on 1, 2, 3, 4. These are the 4 pumps. You collect the sweat under this particular point, and then you push the pump, and that pump then makes the fluid to flow in this 4 different channels and be collected in the radial designs there. And you can do multi-analyte sensing of 4 different analytes. And not only are you doing of the 4 different analytes, you're also doing it 4 times. That also helps you confirm, and that also shows how reproducible the tests are. I know it's epidermal microfluidics, epidermal wearables, so some of you might be falling asleep. I don't blame you for that. Okay, and also we've had long sessions before this as well. Okay, and this is the 4th one here. This is a study that was done by Baig, and this was published in Science Robotics. Nature and Science happen to be the most popular magazines, very popular journals. We've got very high impact factor journals. And what he has done here is, instead of using an external power source, he's actually used a biofuel powered source which runs on a lactate sensor. So it uses the lactate in the body to power the whole series, the power the circuit, and he's used that for doing multiplexed and wireless biosensing at human machine interfaces as well. So another very good example of how epidermal wearable devices can be used, this time generating its own power to power its own biosensing device there. So to contextualize these, what are we doing in our lab? We call our lab as SSRG. It's got nothing to do with my name. It's Smart Sensors Research Group. We work more on subdermal wearable devices, and the subdermal microneedle sensors are mainly meant to target interstitial fluid, not sweat. So that's how we're doing it different. We are also using AI enhanced. So if you look at some of the work that has been done in these papers, they don't mention a lot about the machine learning and AI, but they are. Baig is also using AI, machine learning and AI. Joe Wang is also using that. We normally use that for artifact correction and for continuous data, and we've also used ours in testing in humans and ambulatory livestock as well. We don't claim to be better than the epidermal wearables, especially if you're talking about glucose sensing. We say the sweat sensors, we can use them as complementary methods for multimodal testing as well. So if you look at the image I showed you here, you've got the circle there with 4 different sensors there. In our case, we use microneedles which have got different islands with 4 different arrays of microneedles, just to kind of put things into the context there. So again, we can use it in, and that's what we call as using it in multimodal diagnostics as well. Multimodal in the sense that you can also have a electrochemical mode of detection, and you can also have a colorimetric mode of detection there as well. Okay, so coming to the future outlook and challenges, well, I think if you want to work on epidermal wearable sensors, the things that you have to mainly focus on are how you can standardize sweat plasma correlations. I mean, that's going to be a big problem, because even people who are working on epidermal sensors, you have to make the person sweat. It's not always possible to make a person sweat unless you have getting the person in ambient conditions. Even room conditions, you can't do that because you have the AC running. I think anybody is hardly sweating here, but as soon as we step out, we start to sweat, right? But then clearly we'll get different values sitting inside the room and outside the room. So we need to have a good correlation study where you can look at the amount of sweat being generated in response to the changes in temperature there as well. So we need some standardization there, which will allow us to then correlate the sweat with the plasma as well. Sweat stimulation methods. How do you stimulate sweat? I mean, you can make a person either exercise, or the other thing is you can use iontophoresis. We saw how a glucowatch was using iontophoresis to extract the sweat out and get the sensors to and get the sweat out and be able to do measurements in the sweat there. And then we also need we've also seen how we have, see, looking at the Baig paper there, where he was using a lactate based biofuel cell, you can improve the power autonomy. When you say power autonomy, meaning that you're trying to have a self powered device which can then power its own biosensing unit, because you need to apply voltage, you need power sources for that, and this can allow in improving power autonomy, and also most important is wireless range. And the other important thing is data. What do you do with the data? How many of you are happy for your data to be uploaded onto into an iCloud which doesn't have any security arrangements, right? Nobody would want. I don't want you to know what my heart rate is right now. How many of you are, no matter how young you are, unless you want to boast about your heart rate being very low, even when you're exercising, you don't want to display, you don't want to share your heart rate. So this is all personal data, and this is a very important problem that we need to understand. So sometimes when you apply for grant funding, they ask you, how are you going to store your data? What about data protection? How are you going to, what are the regulations around your data protection there? How much of this data is going to be in the public domain? People would not want it to be in the public domain. I showed you images of doing clinical studies, and I covered the image of the participant. The reason there being, the participants do not want to be identified. Sometimes you see images of babies being displayed. That's completely unethical. If you're showing images of babies, you have to cover the faces, right? So how do you improve the power autonomy, and how do you improve the wireless range? And to this I would also add about data security as well. Right? How do we make sure that the data is secure? And then add on to this comfort, biocompatibility, and the user compliance as well. I think that's probably my last slide, and we can now open the floor for further discussions on what are the pros and cons of sweat versus interstitial fluid monitoring, what material innovations enable flexible biosensors, and what are the barriers to adoption in clinical settings. We can have this discussion on these three important points. I mean, I have put my case forward. I think epidermal wearable devices are fantastic, right? If I would not introduce you to both epidermal and subdermal wearable devices, I wouldn't be doing justice to you, because I want you to understand both these fields, both these areas, and then choose the pros and cons, and then make your own choices. As I said, there's no competition going on between these different types of wearable devices. They are complementary to each other. Some are giving you good information. If you have a device which is just in the form of a patch which is sticking to your skin and giving you 4 readings in a colorimetric manner, and you can quickly see whether your urea levels are high or not, you wouldn't mind that. I think it depends on your requirement, right? As I said, classic example, pregnancy test kits. The previous versions of pregnancy test kits were ones which would give you a yes or a no, answer, right? The new ones are the ones which tell you to what extent, right? How many weeks as well, right? So it all depends on what is the level of information you require, right? I think with that I'll open the floor for discussions. If you have any questions, we can have a chat about the pros and cons of sweat versus ISF monitoring. I mean, based on what you've already been presented, how many of you would actually venture, if you had a choice between epidermal and subdermal, how many of you would venture into subdermal, and how many of you would venture into epidermal? Let's do a show of hands for epidermal.
Yes, epidermal devices. How many of you prefer?
Very easy to make. If you're doing a PhD on it, I think this will be an easy ride, because everything gets much more easier. Your research ethics approval gets much more easier. You're not penetrating the skin. You're sitting on the surface of the skin. You can get some information there, and you can use it in both multimodal manner. You can use it for electrochemical and optical sensing. How many of you would go for a PhD on epidermal wearables?
We can have a show of hands. No worries. Do you understand epidermal sensing, right? So what sort of analytes can we monitor in epidermal devices?
ग्लूकोज स्वेट से और लैक्टेट जो बताया.
Okay.
Sir, epidermal wearables can be used for a wide range of age group peoples.
Okay.
Because for babies, the small children, like epidermal can be a good option rather than the subdermal.
Yes, absolutely. I mean, for babies, epidermal sensors, if they prove it will mainly depend on the function. But if you're trying to monitor urea in babies, perfect. Just stick a patch on the baby, right? You don't need, there's no need for the needles that, I mean, have you ever seen a baby in a neonate unit? Oh, it's a painful scenario. I mean, I wanted to become a pediatrician when I was growing up, but after I joined Liverpool, I had a chance to go to the Alderhay Children's Hospital, and I looked at the babies there in the neonatal unit. Not a pleasing scenario. Babies look cute when they're smiling and but if you see babies crying, it's not an ideal case scenario. Or babies in incubators. Yeah, so it's a good example you've given. Yeah, so you're absolutely right there, that if you would easily get a research ethics approvals if your epidermal wearable device is capable of giving you the information for a baby, because this epidermal could be classified as completely non-invasive, right? You're not doing any damage to the skin. Subdermal would be minimally invasive.
Okay, so we can go point by point. So what do you think are from your side, from the audience side, what are the pros? We can list pros of sweat monitoring. So from your understanding, you can tell us. So
what do you think
are the advantages
of sweat? Yes.
It is easily
Well
available.
done. Good answer. Easily accessible, right? You can generate sweat easily. If you want somebody to sweat, just increase the temperature of the surrounding and you can get the person to sweat, right? And then if you want to collect it, you can use a microfluidic device, a very, very basic microfluidic device. Even a paper, you can even use a Wattman filter paper to collect sweat. It's like collecting tears. There's a strip called a Schrimmer's tears because you get very small volumes there, right? That's good. That's the advantage of sweat. What else?
So as a target analyte fluid, it's easier to collect it. Yeah, anything else?
Actually,
sit, don't worry. Yeah.
the sweat is readily available in the human body.
Yes.
Already there, and also we can use in the region like India's, because it is near the equatorial line, so there the temperature is high, so the the person has the more sweat concentration rather than the region in the western countries, so it is more useful for us also.
I agree, but just to make a point very clear, I think the sale of deodorants is not very less in the western countries than in India. People there also sweat quite a lot. It's just that it's not very evident.
And also cons is basically the concentration of the glucose.
Pardon?
The problems of the sweat uses analytics because the concentration of the glucose is not prominent, or it is variable in the sweat, so we cannot use.
Yeah, I agree with you on that.
So
I'll take that point as well. But what is the bigger problem there? There's no good correlation. Exactly.
Yes, there is no good.
Good. I think we can move to the ISF now.
Just coming to just
Yes,
one
please.
minute.
The quantity of sweat can be also an issue because some people sweat a lot and some
Agreed.
are also having a genetic condition where they excessively sweat,
Yes.
but others have different.
Yes. People who have problems with their hypothalamus in the brain, they have this condition wherein basically they sweat profusely. Just eating makes them sweat. Just sitting there quietly makes them sweat. Have they switched off the AC, or are we testing the sweat-based sensors? I thought somebody's trying to make me sweat now.
Sir, also another problem is like in the winter season the person does not sweat a lot, and in the summer it
Yes,
it
agreed.
is time dependent also.
Yeah.
Okay, what I also wanted to mention is regarding the correlation. Okay, it's not very strong that it justifies, let's say, glucose. The correlation between blood glucose and sweat glucose is not strong enough to justify a diagnostic application. But there are certain applications where you just want to get an idea.
Yeah,
It's
point measurements.
point measurements.
Agree.
It
So
can be
more for point of care kind of things rather than for continuous monitoring or for having it as a wearable device. Because wearable devices, as we say, something that you wear for a particular duration, and that duration could be anything ranging from 4 hours to 6 hours to a whole day. Or I think that we'll do that. What I'll do is at the end of this session, I'll show you a continuous glucose monitoring sensor and I'll implant it on myself.
okay.
And then we can just kind of get some idea there as well, and then it's going to sit there for 14 days, but I'll send you a picture which you can circulate to show what happens when you remove it outside. Show you a little bit of blood there as well, to show you exactly how deep it goes in.
Yeah, we can do that. So we have some continuous glucose monitoring sensors, and we also have microneedles. We have PCB based sensors that we can show you now in the next session, as well as in the during the hands-on experimental. So compared to sweat, now like for example, I guess has anybody done a comprehensive study of what sort of analytes that can be monitored in sweat? So in ISF, of course, it's the so in ISF we find most of the analytes that are present in plasma we are able to detect in ISF as well. What about sweat? So in for example, in ISF the main issue is the size of the molecule. The smaller the size, the more available they are in the ISF. Is it the same in sweat, or how does it? What is the link?
Sweat is normally coming through the sweat glands.
Right.
But having said, so you normally expect to see metabolites there. That's why urea is very common, glucose is very common, lactate is very common, alcohol is very common.
In sweat.
I mean, you guys are this thing. So when people who are excessive drinkers, even their sweat stinks of alcohol. You can actually, if you monitor their sweat, you can even get alcohol from their sweat as well. So because that's a very small molecule, ethanol, C2H5OH is very small. You can easily calculate. You can see it there as well. You can see ketones in sweat as well.
In sweat. Okay. Interesting. Yeah. Any other points you'd like to make?
Again, one more thing I'd like to mention is this: when you're close to the sweat glands, you also have certain hormones being hormonal gland ducts as well. Cortisol. But the only problem there is that there's no good correlation between these cortisol and your the hormones that's produced, even the reproductive hormones and blood. That's why people are not using that for those kind of studies.
So we may not be able to use it for like therapeutic or diagnostic applications.
No, it's more for well-being kind of thing.
Yeah, it's kind of well-being, digital wellness or kind of wellness where we want to get a rough idea of health.
Yes.
Without going too invasive, we can still have some idea from the sweat monitoring device. So again, in ISF, I guess ISF is a very good fluid to like use for monitoring applications, for continuous monitoring applications, I guess. So so in terms of so that is a pro for ISF, that it is having strong correlation with blood analyte levels, so you can use it for many applications. So if you are developing something for a diagnostic or therapeutic application, then ISF would be a better target.
Absolutely.
And if you are doing something just to get an idea or sort of which has some wellness application without any direct health related application, then we could go for sweat, I suppose. So moving on, so many people have developed these flexible biosensors. So what would you say is the challenge remaining to be overcome in flexible biosensors?
I think what we need there is, so what's happening is we are still sticking to polymeric materials. I think we should start looking into natural polymeric materials.
Which are biodegradable.
I think that green, somebody mentioned green chemistry yesterday. I can't remember, it was a gentleman who mentioned about green chemistry, spoke about green chemistry. So if you're talking about green chemistry, because I mean, we are actually going through a lot of, this pollution thing that's going on around. So the AC started again. I think as soon as we move to ISF, they've stopped making me sweat now. So basically, I think it's all about biocompatibility, and it's all about the environmental side of things as well.
Okay. So do you have any so what are the typical polymers that people are currently using? So polyamide is one.
PSS and all these are all materials that are being used. Polyamide, PET, Ecoflex, these are the materials that are being used right now. At the moment, people are not worried too much about about the environmental side of things, because this is research in early days. It's only the concern will be when you start getting multiple of these devices.
Okay.
To wrap them, you need, so I think silk would be a fantastic polymer because it's a
Silk,
natural
yeah.
polymer. Cotton, paper,
Okay.
anything that's natural polymer, and then inks that you use for them.
Okay. Nice. Now it's good to know. So, and okay, moving on. So, what are the barriers to adoption in clinical settings? What would you say, doctors, from your experience?
I mean,
Uh,
I think the gentleman there gave a perfect example on babies, you know, so neonates. I mean, nobody wants to see, you know, already these, you have so many things being already kind of, plumbed into the bodies of these neonates. The last thing you want to see is another device which is again a bit minimally invasive or invasive to go into these neonates. But if you can have something that can be stuck to the diapers, so that you can monitor from sweat, or we can monitor in urine and stuff like that. That's perfect. So the barriers to adoption is epidermal microfluidic devices or epidermal wearable devices would have lower barriers for clinical settings. Anything that's invasive or minimally invasive will have slightly higher, and not slightly, will have higher barriers for adoption in clinical settings.
Okay. And these are which class of devices? Like we were discussing yesterday, just to recap.
That one would be again still be considered as a class 2 device.
Class 2.
It depends on the
From-
material that's being used,
from a
because don't
regulatory-
forget polymers in contact with skin,
So, from a regulatory perspective, it would still be class 2.
class 2.
Right. Okay, interesting. So, okay, so we have few more minutes, so we can actually any suggestions or any points you want to add here from your side?
Sir, talking about the ISF fluid.
Yeah.
I have just one question. Is the concentration and composition of ISF is all around the same in our body? Because it can vary, sir.
I mean, the skin is, as I said, uniform. The skin thickness is uniform all over the body.
Okay.
And therefore the assumption is that the subvascular plexus, the distribution of capillaries, is also uniform all over the body.
But I don't think, sir, it will be uniform composition.
You're talking about the superficial. You're talking about the extremities of the body. See, you shouldn't be confused between distribution of blood vessels in the organ and in the skin. Skin is definitely an organ, but the distribution of the subvascular plexus is much more. So if you're looking at, if your assumption is based on the color of skin being different in different parts of the body, that's related to your melanocytes. So the melanocytes area which are exposed to the that are more exposed, that's part of the skin is more darker in color. That's not got to do anything with the blood distribution. The subvascular. So what I'm trying to say here is the ISF is in constant equilibration with the subvascular plexus, the very fine capillaries, right? And this distribution of subvascular plexus is, at the moment, the hypothesis is that it's constant all over the entire body. So if I did a measurement on my forearm or my back arm, it shouldn't make a big difference. It won't make a difference.
So, in this PPT, do you have, just to add to his question, like, or I would, so do you have an anatomy of the skin in this PPT?
Unfortunately, no, this is more on epidermal, so I didn't go into
Okay.
the anatomy of
No
skin.
worries.
But we can discuss that when we should be having subdermal coming up again at some point.
Yeah.
We will discuss it then.
So actually what happens is, you have blood capillaries going all over the skin, close to the surface. So the analytes which are leaking out of the capillaries, blood is composed of two parts, right? Plasma is one of them. So whatever analytes is leaking out of the blood capillaries, it's going into the interstitial space. So this is a space which is, the fluid is actually surrounding the cells. And so as long as you have capillaries in and around that space, you would have similar kind of, so that's why there is a tight correlation between blood and ISF. It's because of the capillary blood which is going there and innervating these areas.
While the sweat is produced by sweat glands.
Yeah, while as the sweat is produced by sweat glands, and
So things have to actually diffuse from the ISF into the sweat glands.
yeah, and then from the sweat it comes out. Yeah, exactly. So, so are there any ways to artificially produce sweat, like by stimulation current?
There are different ways. I think if you did some literature review, there are different ways of getting artificial ways of producing sweat.
Okay. So there, yeah. I think there are some materials like some chemicals also which
Yes,
can
yes.
stimulate. Yeah, there are also other like papers I have also seen in which people are using.
So for all
various
these studies, they have actually stimulated sweat. It's not natural
So-
sweat.
So
I mean, in the US, imagine the winters are very cold.
yeah,
So it's you hardly get anybody to sweat on the surface. You can get sweat around the armpits,
Yeah.
but around the surface, it's really very hard.
yeah, and also you develop devices for all seasons. It's not for only summer that we make the device. So you have to have some artificial way to
Yeah.
stimulate the release of sweat. So is there any link between this reverse iontophoresis and sweat analysis? So can you use reverse iontophoresis for sweat-based applications?
Reverse iontophoresis is basically getting yeah, it's, as I said, even the sweat is a diluted interstitial fluid.
Yeah. Yeah.
Right. So you have your sweat glands which are producing the sweat, which is basically liquid, and it's carrying the metabolites that it's getting from the skin. And where is this coming from? So again, as I said, sweat is more a diluted version of this thing. So that's
depends.
yeah. So and when you do when you do the iontophoresis side of things, you are actually pulling out the ISF, not the sweat.
Right. Yeah. Because I remember seeing some papers where they are claiming that through reverse iontophoresis they are extracting sweat.
Yeah.
But I guess
Well,
it differs. We don't know exactly what it is.
yeah, it's a concentrated version of sweat.
Yeah, absolutely. Right. So so if we were to summarize, so first you have to look at the application that you are working on. So if it is a very health related diagnostic and therapeutic application, then ISF is a better target fluid than sweat. And then if you're working for something where you want to get a rough idea of like from a wellness perspective, then you can use your sweat as well. And there are many ways to extract the ISF and the sweat. So ISF generally, we, like sir was saying, we go for subdermal devices, minimally invasive and easy to access the ISF. And there are flexible biosensors that are available, and flexible biosensors have many advantages. And the future is, as sir is saying, is to look at polymer materials which are biodegradable and good for the environment. And the barriers to clinical adoption are, of course, as you move from a non-invasive to minimally invasive device, the barriers increase, and you end up in the class 2 categorization of the regulatory side of things, and then you have to meet all these regulatory approvals. So I think that's all for this session. We will reconvene again in the next half. We'll let you know the timing.
Great. Thank you.
Start. Okay. Good afternoon, everybody. So we are continuing our GIAN lecture series, and in this today afternoon we would be discussing more about a lot of other point of care devices, majorly optical and other challenges associated with these point of care devices, and what are the design challenges primarily. Okay. So, if you look at the anatomy of the biosensor, which has been already discussed by Professor Sanjiv, we can see that there are a lot of different type of the samples are being used, and he has talked about the biological elements like antibodies, aptamers, enzymes, MIPS and the other types. Apart from that, other party includes these transducers like the electrochemical transducers, optical transducers, calorimetric and others, and then whatever the signal is being developed or being transduced, these signal need to be processed to be displayed or to be given to an individual who is using these biosensors, right. So, sample is the one part, in between lies the entire sensor, and at the end there is some sort of the readout is there. Okay. Now, these samples are very important. So, all the while we were discussing about wearable biosensors, and in the wearable biosensors it need to be either worn on the different parts of the body, it should be contact with the skin, or it should be under the skin in the dermal area like the microneedle sort of the biosensors. So, these are being accessible to the type of the biofluids, primarily the sweat, or it could be your interstitial fluid. However, one of the major biofluids or the sample is the blood, and which is still considered as a gold standard for measuring lot of analytes of the diagnostics. So, that is why using this blood as a sample is also a primary source, and the lot of the innovations people are carried out, which we will be discussing in the further slides, where blood as a sample is being used, but making a wearable where it stands the difficulty, right. So, for example, in the glucose sensor, which we have Sanjiv have already discussed that for the diabetic measurements, you have these strips where you put a drop of blood from your finger prick, and this finger prick, this blood traverses through the channel and reaches to the electrode area and then gives us the signature, right. And many a times we might have observed while using this sensors that when you put a drop of blood, it tends to get clot, and every time you have to take the new sensor to get the reading, and couple of get wasted in the process as well. So, it is primarily because the blood as a fluid is very much different from any type of the simple Newtonian fluids like the water or analyte mixed in the water, right. Now, if you look at this device, this is the lateral flow device, and which is being commonly used, or one of the most prominently diagnostic tool which is being developed and used by everybody. For example, pregnancy kit is the type of the lateral flow assay device, and if you look at the lateral flow assay device, here is the sample, and then in between there is the sensors or the antigen or antibodies are there, and then the fluid interacts with the antigen and antibody, and then it generates some sort of the signal in terms of the color, this is calorimetric sort of thing, and then based on the color we identify whether the analyte is present in the sample or not. Okay. But if you look at entire thing, the first part is the sample part, where the sample need to be deposited. So, for example, if you look at the urine where the pregnancy test is usually being carried out, it is a Newtonian fluid, you have a lot of samples, but if you take the prick of a blood and put it over there, it might not flow through the entire device, and in between it get dried up or get clotted. So, this creates another challenge of how to drive blood sort of the samples through this sort of the porous matrix. Okay. Now, when it interacts with the test line or the control line, then many a times the blood is having this color, red color. So, the signatures or the signal which is likely to come out may not be visible for the optical readout if you are using the blood. So, these are the few challenges with using samples like blood in the lateral flow devices. Now, apart from the blood, there are the n number of other type of the biofluids are there. For example, the saliva is there, sweat is there, tears are there, cerebrospinal fluid is there, follicular fluid is there, urine is there, umbilical fluid is there, synovial fluid. So, all these fluids are having different types of analytes, and accessing some of the fluid would be easy, some would be difficult, but having all the markers or the analyte in all the samples, all type of fluid, it is a difficult. That is why we need different type of the biofluids for the different type of the analyte detection. So, now if you look at the example of the blood, we would be talking a lot about the blood and how it could be used for the point of care diagnostic tool. So, major method of the blood collection is arterial sampling or the venipuncture. You must have all undergone if you have visited any diagnostic center, and this is common practice for the adults. But if you look at for the infants or babies, they insert a needle in their forearm and then collect the blood through this method, and this is quite painful. So, now when my baby, I had a kid and she was at 1 year old, then I could see that the doctor used to collect the blood like this, and it was quite uncomfortable feeling for a parents to see the baby crying and the blood is just dripping down for any analytical tests. Apart from these things, if you look at the blood as a sample, it is a very fragile sort of the fluid is there. It need to be stored under the cold condition, like the cold chain transport is needed. So, if the blood is being collected, it can be stored into the cold conditions. Then blood need to be processed before it could be used for the analysis purpose. And moreover, if you look at go to the any diagnostic clinic, you will see that 5 ml of the blood is generally being drawn out of your body, even if you require only 1 ml of blood. So, there is a lot of the wastage of the blood is also associated over there, right. So, this method of the blood as a sample collection is not very efficient or not very good. So, people have come up with another method. So, for the babies or for geriatric patients where the repeated sampling need to be done, one of the best is the dipstick method. So, here you can see a capillary is being used, a lancet is being used to prick the finger, and then the capillary is used to draw the blood, and similarly the glucose testing can be done through this, and this is the type of the lancet we generally use to prick, right. Now, many people fear the lancet as well for getting out of the blood, but that is the best possible minimally invasive sort of, or invasive but not too painful or causing a lot of the discomfort. For example, for in the case of the babies, if you look at over here, from the heel prick area they collect the blood samples and they collect it on the FTA card. So, this is a microporous paper sort of thing is there, and then you collect the blood drop in the dried format and then you use it for the analysis. So, many of the genetic screening or the bilirubin test and other test is being done through the heel prick method to identify whether the infant who is born is having some sort of the genetic disorder or any other disorders. Now, if you look at entire this FTA card, this is the how the specimens are being collected in general, right. Here you see that the quantity is not sufficient. So, you have to do the repeated pricking, which is not at all desirable to do repeated pricking of a at least an infant, at least in adults or the grown up can bear it, but repeated pricking of the infants would be highly discomfortable. Wet specimen, So, here the specimen is not correctly being put over in the FTA card. Here you can see the supersaturation. So, lot of the blood has saturated and clotted over the region. Here the contamination. So, there is a specific way so that the blood only touches the FTA card and this sort of this entire area is being completely covered with the blood, not it is being touched by the heel or finger. Here the serum rings are created. So, these are all the examples of the invalid samples. So, when sampling is being done for the blood, these sort of the invalid samples are primarily being seen. For example, here you can see the sample 2 and sample 3 is not the adequate one, whereas you need the sample like 3, 4 and 5. Now, major reason behind is that blood is a complex fluid and this FTA paper is a porous matrix, due to which, because of the person to person hematocrit variability. Hematocrit variability is basically the blood cell component of your entire blood. So, blood consists of the plasma and the blood cell component, and more is the in the females it is higher as compared in the case of the males. Can you guess why hematocrit is higher in the females? Or it is higher in the males? What do you think? So, it is basically the requirement of the blood by the male or the female, right. So, one thing is that the whosoever blood requirement is higher, oxygenation requirement is higher, the blood cells need to be higher, right. So, hematocrit volume will vary depending upon the type of the physical activity a person does. Then it also depends upon the blood volume, like somebody is not well hydrated, somebody is hydrated. So, that also governs it. Then the viscosity is the another issue. When the viscous fluid moves through a porous matrix, it tends to clot, or due to the shear the blood cells led to blood cells tends to collide with each other and then clog these pores of the porous matrix, hence the flow of the blood is being disrupted. Then as the blood is red in color, the difficulty in the visual assessment. So, using blood as a whole as a sample is a difficult, that is why in the clinical setting people use the plasma as a blood, plasma of the blood for this sort of analysis. So, for that the plasma and the blood cells need to be separated through the centrifugation or any other method. Hemolysis is the another issue. So, reason behind hemolysis is that to massage the finger or the heel area before being blood is being taken out for this FTA card based. Now, in that case there is the chance that it mixes with the interstitial fluid or the blood cells may get ruptured over there and you may not get cell. And then the sample preparation complexity, this is the another issue which is says that after the blood is being taken, it need to be processed to remove the cells, then take out the plasma or to take out different analyte and different type of the solvent and then use it for the analysis. So, the complex sample preparation is the one of the important issue. So, because of these 5 factors, using blood and the POC like LFA devices is a challenge. So, if you look at the different blood micro sampling techniques, so people have come up with the different ways how you can sample the blood apart from this FTA card. So, for example, this is the VAMS technology, volumetric sampling. So, if you look at here, a prick is being done and then this sort of device is having this porous matrix which is able to absorb from the prick without being touching the finger. So, contamination issue can be avoided. So, similarly, this is the another sort of the device which is being worn over here. So, it has a needle, it will insert inside and the suck like a mosquito, right. Now, these are the different ways people have tried out. Here are the another ways, like people have created how to separate the plasma and the blood cells, so that the post processing of the challenge associated with the blood samples can be taken care of. This is the similar device, this one. And apart from that, people are still innovating on the FTA card itself, that how they can manipulate the fluid flow in a FTA card, so that uniform spreading of the blood can happen. So, for example, they have created this sort of the structure, and this structure lead to the uniform spreading of the fluid. So, by altering the geometry of the FTA card itself, one can achieve the uniform spreading as well as the flow of the fluid. This is the another technology in which the blood is collected in the spiral format, so that uniform spreading can be attained. This is the another technology through which people are trying. So, lot of the innovations are being going on to how to sample a microliter volume of the blood for different analysis. Now, if you look at this blood micro sampling itself, there are a lot of articles were published in 2022 only, and this had a lot of applications as well. So, for example, this micro volume of the blood is a very important. So, in the morning Professor Sanjiv talked about the PK/PD studies. So, in those studies, let us suppose a patient is having a cancer or some immunosuppressant drug, and that drug need to be evaluated, how the person is performing when a drug is being given, how it is being metabolized through the 24-hour or 48-hour period. So, in such case, many a times currently the practice is that 2 ml of the blood is being taken every 30 minutes, and they imagine the condition if the person is a geriatric patient or an infant. So, those sort of the condition creates a lot of issue. Now, getting a small amount of the blood and then doing all this analysis is a very important, and here comes the this sort of the technology is called micro sampling technology. So, it achieves the 3R policy. It enriches the PK/PD data, because you can sample a small amount, not 30 minutes, but in the 15-minute time, and you have a enriched data of the PK/PD studies. So, you can have longitudinal study for long without causing lot of discomfort to the patients. So, this has got lot of applications like therapeutic drug monitoring, neonatal and pediatric studies, animal studies, biomarker research, forensic technology. So, these all, and due to this has led to the minimally invasive, self sampling is a easy accessibility, and all this collection has reduced the resources, improved analyte stability, and facilitated the automation process. So, for example, a small amount of blood being taken by VAMS technology, it can be inserted inside a automated machine, and it can do all the analysis. So, that is quite easy. So, towards this we also worked, and we have developed this sort of blood micro sampler technology, and this is user friendly, biosafe, and the patient comfort. This has been developed in our lab, and this can precisely sample the blood 30 microliter, 50 microliter, 100 microliter, because if you have the defined volume of the sample blood, then you can do the quantitative analysis along with the qualitative. Otherwise, you have to restrict only to the qualitative analysis, and many a times during analysis we need quantitative analysis. So, and this can be used for any biofluids. We have tested it for the saliva, for the blood, urine. In all these cases, it is working fine. So, how this device is? This is the holder, and this is the porous matrix, and it is a simple similar to that of VAMS technology, which I have discussed, but VAMS is not Indian technology. So, this is the first time we have developed in India, and this porous matrix is the specialized hydrophilic porous polymer matrix, and it is able to suck the defined volume of the blood. So, by tailoring the hydrophilicity and the pore size, we can tune it for making the 30 microliter, 50 microliter, 100 microliter, and this sampling blood, when is there, it can be dried, and then it can be transported without the need for the cold chain transport. and anybody can use it for the home sampling, and then the blood would be transported to any of the pathological chains or the diagnostic center where it can be retrieved for the analyte detection. So, this is the final device which we have come up with, and this has already been approved in India for clinical use and ISO certified as well. So, he is my student who has worked on this technology, and he has commercialized through his company IOTA Diagnostics Private Limited. And we have got certain publications out of this microsampling technology, and which we have shown that it can be used for the pharmaceutical analysis, it can be used for the RNA sample storage, which is very important, was in the case of the COVID-19 pandemic. So, this dried matrix could be used for the storing of the blood, taking out the blood samples, and then storing and sending it to the any across those. So, it has increased the accessibility of the diagnostic for people living in the remotest of the country. Now, if you look at the blood samples, one of the blood samples is the menstrual blood also, and the menstrual blood is very difficult to access because sampling is a big issue over there. So, for example, if you look at the cervical cancer cases in India, it is a continuously rising, and even the government has launched the cervical cancer vaccines, and to facilitate this cervical cancer screening, female has to undergo pap test. That is the molecular diagnostic test one has to go through, and this is very difficult because many of the people are unaware also because of the social stigma, the culture around. So, difficult to go for the pap test because the swab has to be taken from the vaginal area, and many people are not comfortable with it. As a result of which, we have seen that the cervical cancer is consistently increasing. But there has been a report which says that the cervical cancer can be, if known, it can be treated as well, and it can be diagnosed early with the help of the menstrual blood. Now, the challenge was previously to how to collect those menstrual blood, because it is still a taboo in India and in many geographies where people feel that it is not safe and all those things associated with it. Now, facilitating the self sampling was one of the innovations which we have done at IOTA Diagnostics. So, we have created a strip called the M strip, and this is the sanitary pad. So, we have created a device which can be integrated over here like this, and then it could be placed over there and it could be worn by the females, and then after the periods this blood which is coming out during the periods can be collected in these sort of the samples, and then these samples could be put it inside and the couriered to the given laboratories for the diagnosis. So, it overcomes the personal hitch or personal society related taboo of sampling. So, in micro sampling or the blood sampling, it is very important that one need to consider what type of the geographies you are associated, what sort of the samples you are collecting, what type of fluid you are collecting, because all these will drive the level of the innovation or the design of your device. For example, here it is a device something like this, having this porous matrix attached to it in a way that it could be applied on this strip, whereas in the blood microsampler it was a pen sort of the device. So, the design of all these devices will vary depending upon the type of the fluid you are sampling and in what place, who is your intended customer, what is their paying capacity, all this governs the design of these devices. Now, apart from this blood, as these two examples which I talked about because I had worked on it, there are the n number of other fluids which we have already discussed, which are difficult to access, but they have the analyte which need to be detected. So, you can think of different innovations. So, for example, the tear is the one of the fluid. So, this is the how the tear is being collected. You take a capillary and then put it inside the eye, and you collect the tear and do the diagnosis of this. I do not know if by mistake if your hand flickers, then you may poke the eyes of a person. So, that is or there is this test, shimmer test, where this is the porous sort of the paper is there, and where you put it inside and tear just down it, and then it is being collected, right. So, can you think of a better way of collecting these tear as a samples? Pardon? Okay. So, it will come to your cheek and then then it may get contaminated because of the cheek analyte or sweat, right. So, similarly, this is the another device which has been created for the sweat sampling. So, sweat is common in the India and these geographies, but it is not common in the, I think, in the UK and the near the poles the countries are there. So, how do you create the sweat? To collect the sweat and then do the analysis over it. So, people have created this sort of the absorption pad is there, then the patch is there, then it collects the sweat slowly, which get accumulated, and then it can be used for the n number of analysis, right. So, the message I wanted to give it from this part of the is that there are a lot of the blood biofluids are there, and each biofluids have a specific diagnostic applications. All cannot be accessed with the help of skin or sweat, but they need to be taken out and do you can have a point of care diagnostic tool associated with it. So, for that you need to sample those analytes and then only you can use it. Now, another part which I found during the course of this GIAN course was the biosensors, where some students were talking about the optical biosensors, and why do not we develop optical biosensors as a wearables. So, this is very important to understand what these actual optical biosensors and what are the challenges. Few of the challenges Professor Sanjiv had listed in the morning, right, related to the cost. So, we will talk more about what is the exact what is the component which causes the cost, why it is difficult to develop, what are the other challenges associated with it. Okay. So, before moving to this optical biosensors, this is the biosensor, optical biosensor which we all are familiar with after the COVID-19 pandemic, right. So, this is the pulse oximeter, and it measures a very important vital signature, and which is the oxygenated blood. So, it's rapidly estimates the peripheral oxygen saturation and provide clinical data in a very efficient and non-invasive and the convenient manner. So, now if you look at the principle, here is there is a one module is there, and here is the another module, right. Now, in this module they are having one source and one detector. So, your source is like this, then your finger is like this, and then your detector is like this. Okay. So, now you need some source and you need a detector, and then whatever the detector is detecting, it is being transmitted and then it is being displayed over here, right. So, you need a display unit or tell you what is the amount of the analyte present during the optical detection. Now, here if you look at here, we use two types of LEDs. So, LED is used as a light source over here, but these LEDs are of the precise wavelength. So, one is the I think 660 and one is about I think it is 940. Okay. And if you look at the HbO2, the red one, this is the oxygenated one, and the blue one is the deoxygenated one. So, based on this ratio and this ratio, whatever. So, when the light passes through your finger is having muscles, then bone, all those interference is there, along with the blood is also there, right. So, without blood, what is the signature? That is taken as the base level, and with blood, what is the base level? So, based on that absorbance, it is being calibrated, and based on the ratio of the absorbance at two wavelength, we measure the oxygenation concentration, and we represent in the form of a percentage. It is a relative oxygenation. Okay. And this is just the display unit. So, display unit can be miniaturized, and that is well established in the literature, but the major challenge lies in this detector and the source. Now, before moving further on this type of the optical biosensors, so from this it is quite, you must be thinking that it is easy to use this type of the optical biosensors for any type of the analyte in your body, right. And that is the mistake which I also did when I in 2010, when I thought of using this sort of architecture of the optical biosensor for detection of the biomarker inside our body in the blood, like for the cardiac detection, like troponin T, right. So, if you look at the electromagnetic spectrum, so let us come back to the basics of how the light interacts with the material and what we are exactly measuring, right. So, there we were measuring the light of a certain wavelength was interacting with the oxygenated hemoglobin and the deoxygenated hemoglobin. So, both are having the different type of forms, that is why they were interacting with the certain precise wavelength of the light in a different way, right. So, how these interactions are there, and are these interactions specific enough, or what is the challenge associated with these sort of the interactions? Because in the case of the electrochemical sensors, if you have learnt, you have seen that you have used the biorecognition elements, and these biorecognition elements provide high specificity and the sensitivity, right. But here it is the light which is going to interact with the material. Now, there are a lot of the different ways or the different wavelength of the light which is going to interact. So, if you look at the UV visible, right, they are having a certain range of the frequencies. If they are having the certain range of the frequencies, they are having the certain amount of the energy, right. From here you can calculate the energy of the any wavelength of the electromagnetic spectrum, and if you know these energies are going to interact with the material or the matter, right. For example, the infrared is having this much less energy than that of the UV visible. Radio waves are having further less energy, right. So, all those phenomena inside a material when it is interacting, if energy is of similar nature. So, for example, you must have studied in your +2 that when light interacts with the, let us suppose, the atom, the electron gains the energy and jumps to the higher energy levels. Now, these all things are quantized in nature. So, this means that you need a specific amount of the energy or a specific wavelength of the light to make the electron jump from the inner orbitals to the outer orbitals. So, that is why these energy levels are the very important. So, certain transitions are only possible with these energies, and certain transitions are possible with these energies only, and certain transitions are possible with these energies. So, that is why different class of this energy of the electromagnetic spectrums are being used for interacting with the material, and then we are measuring the outcome. Okay. So, for example, here you can see this is the Jablonski diagram which Professor Sanjiv was talking on in the morning. So, if you look at this is the S0 energy level, this is the S0 is the ground energy level, and when any of the electron gains some energy h nu, it jumps to a different energy level S1, S2, S3 depending upon the type of the energy it has got. Now, when it gains the energy, some of the electrons tends to come back to its ground state, and while coming back to the ground state, they releases some amount of the energy, right. So, here you can see jumping to the higher energy level and then relaxing and coming to the S3, then so these are the relaxation based energies. These are quite instantaneous in nature, right. Then there is the fluorescence based. For example, it jumps to the higher energy level and come back to its ground state, then the fluorescence is being observed. Okay. And many a times what happens is that phosphorescence is the another type of the this sort of the transition. In that case, it jumps through the another energy level. So, these are called the triplet state and the singlet state. So, for your understanding, singlet is, so each of the electrons exist always in the pair, right. According to the Pauli exclusion principle, it should be clockwise and anti-clockwise, right. So, singlet is the condition when it is the clockwise and anti-clockwise. So, if in the ground state it is in this direction, in the excited state the other would be. So, this is called the singlet. But when the condition happens that in the ground state it is like this and the excited state, it is called the triplet. So, when it happens in the case of the triplet, phosphorescence sort of the phenomena is being observed, right. So, in brief, this diagram just says that a definite amount of the energy is being required to make the electrons jump from the ground state to the higher excited state, and moreover depending upon the singlet and the triplet state, it will show fluorescence or the phosphorescence. And when it is happening instantaneously, relaxation, you will not be observing any visible change. Now, using this, if you look at the transition timing, so this excitation, these are almost instantaneously, whereas in the case of fluorescence and the phosphorescence, you can see the time scales in the case of the phosphorescence is quite large. So, it takes some time to phosphor the material, whereas in the case of the fluorescence it is instantaneously. If you take any of the material and interact with the light, it will show. So, now as we discussed that definite amount of the energy is required to pump the electrons to the higher energy levels. So, visible region of the spectrum comprises of the photons of the 36 to 72 kilocalorie per mole, and the near ultraviolet region of the 200 nanometer extend the energy up to 143 kilocalorie per mole. So, this is having in the UV range, this having more energy as compared to the visible range. That is why we can see some molecules, many of the molecules we detect near to the 400 nanometer range or below it, 200 to 400 nanometer. Now, if you look at any of the organic molecules, so all those materials in our body you can consider as the organic molecules, right. And all these organic molecules, you must have heard about this bonding and the anti bonding. So, SS bonding, SP DF bonding, do you remember from the high school? So, it is basically all these electrons are like the electron cloud. So, atom is there and there is the electron cloud is there, and when it need to bond with the another associated another atom, so let us suppose this is A and this is B, this electron cloud is going to interact with each other. So, there is the electron cloud can be considered in terms of the waves as well, and if they can be considered as a wave, they can have a constructive interference or destructive interference. So, if the constructive interference is there, it is called the sigma sort of bond if you take the s orbitals, and if it is a destructive interference, you can say that sigma star sort of. So, you can see your sigma star and then sigma. Similarly, if the p orbitals are going to bind with each other, they are going to form pi star and the pi. Now, this anti-bondings are unstable in nature. They are so they are easy to knock the electron out of there. Okay. So if you look at these jumps, so there are the pi bonding is the most stable one, and the pi star is the most unstable one. Sorry, sigma star, and in between lies the pi star and the pi bonding, and then this is the non-bonded, like the nitrogen or the phosphorus, one of the electron do not get bonded. So those are the atoms we are talking about. Now there is this, all the jumps are possible, but some jumps requires the more energy, some requires the less energy. So where there is a these sort of the transitions, pi to pi star, sigma to sigma star, these are the quite, but these are not possible, these two less possible, where these are quite possible. Okay. So you can think of any light which is going to interact with the organic material, it is going to show this sort of the behavior. Is it clear? Now using this as an analogy, so you can see that when the pi to pi star is happening, if the energy level of this one is like C double bond O, and this is the pi star. So when this transition is there, it has to move to this. Now when there is the conjugated pi bonds are there, right? For example, like this, here it was the C double bond C, here was the C double bond C dash C double bond C. So because of these conjugations, these orbitals come closer to each other and this energy level is getting decreased. Now there is a greater chance of the electrons to jump from the lower orbital to higher orbitals. So more the different type of the conjugations are there, less would be the energy required to bring about the transition. Okay. So for example, if you look at here, this is the one molecule, and if you look at the n is equal to 5, n is equal to 4, n is equal to 3, this n is the basically the C double bond C, this n, and based on this n your conjugation is going to define. So if the conjugation is going to increase, your wavelength is going to increase, your energy is going to decrease, right. So this means that bringing about a conjugation, so all those biomolecules, if the conjugation is there, then only this would be more UV visible active. So you can think of, are there any biomolecules in your body which can show this sort of the behavior, then only it is possible. Then the second thing is that it depends upon the solvent. So majorly UV visible is being done in the different type of the solvents, and each solvent has certain role, but your analyte and the solvent should not show peak at particular wavelength simultaneously. So you have to choose your solvent and analyte different, but if you are measuring inside your body, your solvent remains fixed, you cannot change the solvent. So you have to think about which wavelength would excite. So in the similar way, you can see that in the plants they have different type of the molecules are there, and they have a lot of conjugations. So that is why we see a lot of color in and around. Now having understood the basics of how the wavelength interacts with the or a light interacts with the molecules, here is the UV visible instrumentation. So if you look at the instrumentation, there are a lot of the components are associated with it. For example, you have a light source, then it need to be filtered, then lens is there, shutter is there, here is the sample holder, lens is there, slit is there, grating is there, diode array is there, detector is there. So all these components need to be aligned, right, because the light travel in a straight line and all alignment issues, and these all are the individual components. So putting it inside a very miniaturized sort of a wearable device, where you would be always moving here and there, it is a very difficult to put it. That is why this is one of the major challenge, that all those optical components are movable in nature, and because of the movable nature you cannot use it as a wearable. So you have to come up with the certain compliant mechanism which is maybe different topic. Through that you have to develop microstructures where each one component would be independent as well as monolithic as well, then only this could be miniaturized. So, people are working on the miniaturizing it smaller and smaller, but it is far away from the wearables, I would say. Clear? So, this is the one of the major reason behind it cannot be used as a wearable. Now, UV is a harmful to your body, it can cause mutations and all those things. So, UV part of the wavelength cannot be used for that reason also. Whereas, if you look at in the UV part of the wavelength, lot of analysis is being done near to the UV. So, that is the another challenge. So, people are working on the miniaturizing. This is the one of the work which was published in I think 2021, and they have shown that by miniaturizing this UV. So, this is also a mini spectrometer. This is the one of the costly component associated with the UV visible spectroscopy. These are the simple one, but this is the costly component. So, the cost wise reducing a optical sensor is a little bit more difficult. So, here is the two examples of the ocean optics, and these are handheld or portable sort of, but far away from the wearables. So, I hope why UV visible, although used for the analysis or analytical chemistry or the measurements of the analytes in the blood, but you cannot use it for the wearables, at least current day technology do not support it. Now, coming back to the another technology which is the FTIR, a Fourier transfer infrared spectroscopy. Here the energy is from the 1 to 15 kilocalorie per mole. So, energy is quite less, and here we are not expecting any sort of the electronic transition, but here what happens is that when the light interacts, it interacts with these dipoles, and when it, so you can think of the light as an electromagnetic wave having the electric field and the magnetic field, and these molecules like the formaldehyde. So, these are the carbon atom, oxygen atom, then the hydrogen atoms are there, and this can act as a dipole. So, when a charged dipole interacts with the electric field, it is getting stressed or squeezed. Now, these molecules are having inherent movement as well, right, and when it interacts with it, it brings about the change in the dipole when the light interacts, and that change lead to the absorption of the light. So, you can see that the transmission. So, if you look at this is as a detector, when the light passes through this molecule, whatever is detected. So, if it is being absorbed at a certain wavelength, the transmission is going to decrease. So, detector will detect less amount, and this is what you record in the FTIR. For example, this one percentage transmission. So, at certain wavelength, these vibrations matches with that of the your electromagnetic radiation which you are signing on it. Okay. Now, if you look at all the biomolecules in your body, it is primarily made of the hydrogen, carbon, nitrogen, and having the bonds like C double bond on, CH, OH, NH, NH2, C double bond O, right. But this technique only detects these individual functional groups, not the entire molecule as such. For example, if you take the glucose and if you take the fructose, possibly individual peaks will match, but they are the different molecules. Whereas, in the case of the electrochemistry where the bioreceptor is there, it can identify between which is the fructose and which is the glucose. So, this FTIR is basically based on the Michelson interferometry technique. So, here a light source is being used, then there is a stationary mirror is there, and then there is the moving mirror, and here is the beam splitter, it directs the half of the beam to the stationary mirror, it comes back, and then there is a moving mirror which it is continuously vibrating at a certain frequency, and when the light pushed from this moving mirror, it couples and then it is detected here. Now, it leads to the formation of this sort of the interferograms. Now, if you look at here, this small d is this distance, and based on this distance you can select the wavelength, and if the change the wavelength you can get precisely at this wavelength scanning the material, at this wavelength, at this wavelength, at this wavelength, and you can scan the material and obtain this sort of the graph. Now here is the stationary part, this is the moving part, here is the coherent light source you require, and a lot of other optical components. So on the lab scale it's look pretty good, but miniaturizing it is again becomes a challenge, but people are working on it. It is not that people are not working. For example, these are the 2 research articles published where people have made this sort of the arrangement to miniaturized and make it as much small as they can. Now, so this FTIR, as I said, it detects the functional group. So you can argue that different biomolecules are having the different functional groups. So each biomolecules would be having this peculiar signature, overall signature in the spectra, right. So this was the hypothesis we also thought 10 years back when we were trying to design this sort of the infrared FTIR based sensor for troponin T detection. So recently I came across this literature where people have developed the infrared spectroscopy device for transdermal assessment of elevated cardiac troponin, and this is their wearable device. Now Sanjiv might have shown you a lot of wearable device which was very small. This is like this wearable. Okay, so anyhow our this sort of the optical device has reached to that extent, but nevertheless they are going to progress further, I believe so. Okay. So these are the 2 very good articles which have discussed how infrared could be used for the troponin T detection. So here you can see they are worn on the wrist, right, and then they have compared it. So let us discuss this, how it works. So if you look at the FTIR, you can see these sort of the peaks at different wave number, right, CO and single bond, amide 1, amide 2 and all those things, right. So in that infrared what they have done, if this is the hand and if they have put their sensor, so you can think of this is the some sort of the prism sort of structure is there, okay. This is your hand, and if a light is being shown from here, let us suppose it is showing the total internal reflection over here, okay. Now this light is going to form some sort of the evanescent wave over here, okay. Now this evanescent wave is having the characteristics similar to that of this wavelength, but its intensity is bit less. Now this evanescent waves is going to interact with all those analyte in the dermal area of your skin, because its extent of maximum it can go up to the 500 nanometer sort of the depth. So whatever the analyte would be present over here could be detected, and through this principle they have tried to make that miniaturized FTIR sort based detection, and they have then tested it with the troponin T taken from the patient samples and through the commercial FTIR, and they have calibrated also. So in this is the which shows the ATR absorptions, and these are the areas or these points are the areas where you can see where your this device has shown the indication, so, stating that this could be used to capture the signal from these molecules. Now based on this they have done some clinical studies, and a lot of AI and ML techniques are they have used. I am not familiar with this, but Professor Khalid would be discussing how these data could be used to filter the noise and arrive at the signal. So in this study they have used about 134 data set, and then they have biochemical validations. So clinical validations they have done with the actual biochemical test of the troponin T, and this is the phenotypic validations they have done. And they have used some sort of the convoluted neural network or recurring neural network, and they have used 134 samples, and at the end just I wanted to mention over here that they have reached the accuracy up to 0.84%, and specificity also quite less in the validation set, which is 0.64. So, it is not that this sort of the technique is going to work for the diagnostic purpose, but they could be used for your what do you call screening purpose, or the first line of screening before a person can go to the diagnostic center for actual testing. So, the key takeaways which you can take from work is, first, the sampling of the biofluids for analyte detection through the POC requires the processing because of the nature of the analyte and because of the device which we are using. Then the biofluid sampling is a challenge due to the culture, geography, invasiveness and many other factors. So, while designing any biosampling technique, you need to keep in mind what type of the biofluid and how easily you can take it. Now, optical biosensors are not portable. So, we have seen couple of examples which were quite bulky and still very far away from the portable. For example, electrochemical sensors are looking like a tattoo nowadays, but in the optical sensor it is like a DeX. So, then the cost of the light source, filter, detector and other optical components is a serious challenge in the optical biosensors translation. You requires a lot of filters, you require the optical source, and the detector is the very precise related to a particular wavelength, and sometimes this cost accounts to around in 20 lakhs, 30 lakhs, 40 lakhs. So, it can't be affordable currently. And then final, in the last we saw that for troponin T detection, there lot of the signal to noise ratio was there, specificity was 64%. So, we have to rely on lot of the computational tools, AI and ML sort of tools to filter the and arrive at the actual data. So, thank you. Any questions?
Yeah, any questions from the audience, please? No. I think you might have some questions. No. Okay, so just a quick one. So, where do you think, in terms of miniaturization, optical sensors are heading? Like, what is the research direction for optical sensing?
So,
what is your
right
opinion?
now the miniaturization is based on the compliant mechanism which I talked about. So, these are the monolithic structure where some part would be flexible, some part would be the rigid, and then all the components can be integrated over there, then only it could go towards the miniaturization. For example, in one of the example which I showed one of the person, so this is the one. So, here you can see the lot of the components are integrated, actuation is integrated with the moving mirror, your waveguide is integrated. So, some integration are there. If they are created on a single platform, then only miniaturization could be possible.
Okay. One more question. Since we have discussed lot of different optical techniques, just for the benefit of the audience, can you just name the techniques quickly in terms of just the name, so that it stays in mind? It's like a summary. So, what type of optical methods that you have discussed?
So, these are the couple of techniques. So, for example, UV visible, then FTIR, Raman spectroscopy, LIPS technology, then absorption spectroscopy. So, all different type of the spectroscopy can be techniques. Apart from that, imaging is another technique, but for that you need the camera, light. So, you must have heard about this ingestible pills having this sensor as well as the light which is going to image the inner, but detecting anything at the molecular level would be a challenge. So, imaging wise is the another area where optical techniques can go to take the surface imaging and then do the analysis.
Okay.
Dr. Prasoon, just a quick question. You showed some very interesting examples here. This is a really good talk. So I wanted to know your thoughts on, do you think that optical detection would sometime in the future really match the kind of miniaturization we see with electrochemical sensors? I mean, I know you mentioned the limitations, but you're the person who is the expert in the room right now, who knows exactly how this whole technology is advancing. So do you think at some point there's going to be some form of miniaturization which will allow it, if not complement, at least compete with electrochemical biosensors, portable ones?
In the case of the optical biosensors, as it is, one thing is that it takes the spot measurements, that is the one thing, and then it's a non-invasive in nature. So last example which I showed, it was just measuring all the analyte below the surface of a skin. So those sort of miniaturization might be possible with the help of like optical fibers. So nowadays a lot of biosensors are being developed with the help of the optical fibers, but the challenge with the optical fibers is that the readout. So in future I believe that the readout from this optical fiber, if get miniaturized or let us suppose mobile shaped size, then optical fiber would be able to miniaturize a lot of these optical techniques, and then it may be a possibility.
Okay. Because I saw in one of your slides the setup using Ocean Optics, and I think Ocean Optics is one of those companies that claim that they would be able to provide you with really small spectrophotometers and stuff like that.
Yes.
So
yes.
do you think that there's going to be a possibility of having some FDA approved miniaturized optical? You're absolutely right when it comes to invasiveness. Then these are basically non-invasive because they're measuring from top of the surface. They don't even, So do you think that you can use these kind of miniaturized optical devices to look into the skin interstitial fluid?
If the fluid could be tapped through some microneedle technology, and then because inside the body, see, optical techniques only interacts with the molecule functional characteristics like the OH or COH, NH2, the bonds or individual molecules. So getting entire as a biosensor detecting the analytes like biomolecules would be still a challenge, I feel so, even if they miniaturize it. So, but one thing I would like to add over here, that if the AI and ML techniques, which in the last slide I found that work very promising, if it becomes too advanced and the robust enough, and we have a lot of the data to support, maybe that data processing can help to identify the biomolecules in future, but that is also a futuristic.
Okay, last question. Is there any link, like for example, can you detect any sort of analyte using these optical techniques, or is there any limitation to which molecules people are targeting? Or
so like L and D form of the any biomolecule. So these optical techniques are very good at that also. So but they are not specific. That the major challenge with these optical techniques are they are not specific to a particular molecules, unless we have an entire spectrum of the signature associated with it.
So maybe you have covered, or I don't know, I may have missed it.
Okay.
So the people use this pulse, what color? Oxygen saturation.
Pulse oximeter.
Yeah, oximeter, right. So there it's based on optical only, right?
Yes.
So do you consider that is a miniature form, or still it
Yeah,
is
that
big?
is quite miniaturized form. You can carry it, you can put it, and you can wear it. But it's based on the very specific molecule, like oxygenated hemoglobin and deoxygenated hemoglobin.
Yeah.
So for that specific, we have people have figured it out. What is the specific wavelength associated with it? They have figured it out how to make the light source out of that wavelength. People have ready detectors associated with it. Now, if this need to be extrapolated to n number of other analyte in your body, any other biomolecules, then you need to have specific wavelength which is interacting with that particular molecule and not getting interfered by the other molecules. So,
maybe,
finding out then detector associated with that will be another challenge. So, it is a difficult little bit.
I really don't remember, but somewhere while reading about this pulse oximeter at some time, maybe 2-3 years back, somewhere I came across that there is a multi-component analysis people do. So you can here, let's say, I think 2 wavelengths they measure, but there people have 6 channels, something like that. There are some like miniature form is available. People can use actually for different analysis. I mean, that will reduce probably this interference from other analyte.
So if you have come up with the what is the exact wavelength at which a certain analyte is detecting, and if you are able to have the detector and source for that analyte, you can very well detect.
No, but I don't know whether you can able to get like chips and all.
It's a difficult, as I said. So in the last slide, I have showed one device. So this is an infrared based IR based spectroscopy device for troponin T detection. Now troponin T is the cardiac biomarker which get released only when one is having heart attack. So these analytes are of the very small quantity, and if they are in a very small quantity, earlier you detected the better it is. So now detecting this small analyte, they have used this technique of evanescent wave based interaction with that material and then capturing it. But even if they have captured the signals, their validation of their device with the external biosensor is very less. So they have used algorithms like the CNN models to filter or understand the data to take out their own signature from the other interferent molecules. So that remains another challenge.
Okay, so we would like to close the session now. I think we have one more session left for the day. So thank you very much.
Okay, thank you.
Okay, good afternoon everyone, and welcome to the last session for today's GIAN program. And thank you very much for your attention and for your attentiveness. So today's last program, actually, the session is focusing on instrumentation for subdermal and epidermal wearable devices. So we have been hearing about different kind of sensors: subdermal sensors, epidermal sensors, non-invasive, minimally invasive. So there is always all these sensors. When you look at an application, it's not just a single expertise which is required. It is actually an interdisciplinary problem, and it consists of it requires multiple skills, involvement of multiple people at different stages of the developmental phase, research phase, and development phase. So today's session actually is the goal of today's session is to provide you with the basic knowledge, irrespective of whatever background you come from, whether you are a chemist or an electronics engineer or whether you are a biotechnologist. At some point, if you are interested in sensor development, the session today will help you understand the basics of instrumentation that is involved. So you won't feel lost. You can at least guide people if you have members in your team. Using this knowledge, it will be helpful for you to, you can speak to an electronics engineer, explain your problem to them better. If you are collaborating with someone, you can even develop it yourself. This instrumentation for sensing is not very complex, so you can also learn on your own as well. So we'll have a look at these, what are the instrumentation methods that are used for different kind of devices. So if you look at, if you look at the typical method by which doctors actually diagnose a disease, so they look at the vitals, and then they look at the biomarkers. So the first evaluation phase of a patient is to look at their vitals. So there are many different types of vitals. Let's say ECG, heart rate, blood pressure, SpO2, respiration rate, EEG. So these are different kinds of vitals that ideally are monitored in a non-invasive manner. Okay, and then these vitals are somehow, there is a link between the biochemicals and the vitals. Okay, so if doctors find that there are some abnormalities in the vitals, that is the first stage of diagnosis, then they would advise you to look at the biochemicals. So they might advise you a blood test, or they might ask you to monitor a certain biomarker. They might ask you to check for certain metabolites, proteins, pH, like many different types of markers are there, which actually indicate or help the doctor understand why a certain vital is having some problem. So why there is any abnormality in the vitals. So this review actually written by Kuldeep Mahato, and he is actually part of this group by Joe Wang's group. So they are based in UCLA. So if you are interested in sensors, then in addition to our expert, Professor Sanjiv Sharma, they are one of the other groups to follow their work. And the good thing about this review is they have nicely actually presented the vitals, biochemicals, and all the different kind of sensors that are required for it. So you have glucose, lactate, cortisol. These are some biochemicals that are typically monitored, and all these markers they can be monitored continuously, and they have some use case for continuous monitoring of these markers. Okay. So they have all these markers. There is a user case. There is a justification to monitor all these markers continuously. So there are many markers. There are many biochemicals which do not require continuous monitoring. A spot test is enough. But these are certain markers for which continuous monitoring is required. Hence, we have to if continuous monitoring is required, then we have some justification to develop a wearable device. If it's just for a spot point of care device, then we don't have to think about continuous monitoring. So moving on, so as we have looked at, so this actually is the epidermal sensors that Sanjiv sir previously explained. So this is actually epidermal sensing, microneedle based sensors. These are subdermal sensors. These are subdermal sensors, which are actually, you can see the needle actually goes up to the dermis at max. It's not penetrating into the hypodermis. So it remains somewhere in the epidermis and the dermis. Okay, and the depth is around 1.3 millimeters. So the thick of the dermis. So your needle would be at least less than 1 millimeter in length to penetrate the dermis. Okay, so that's how, and I also learned in this in our in the GIAN program, during the course of this GIAN program. So the tip diameter for good penetration, for the tip diameter, ideally, do you remember what it is? Very good. So it is around 20 microns or 20 micrometers. So if it is around that, then the penetration is smooth and painless almost. And then you see that what happens is here, as Professor Sanjiv was previously explaining, as the needle is inserted, the interstitial layer, the fluid, it forms a layer around the microneedle, and then you can start sensing. So that is why we have this, you can say, sensor start time. That one you cannot do immediately. You cannot. It's advisable not to start sensing immediately. Once you insert the sensor, you allow the sensor to stay there for some time, allow the layer to form, and then you start sensing. Then you will have a much more reliable and a better result. So this is the case also in CGM sensors. Today we will show you some CGM sensors as well, where once you implant the sensor, up to 1 hour you will not be able to get any reading out of this CGM sensor. So that is the sensor warm up time or startup time. So this is your minimally invasive subdermal sensing. So this again is another interesting technique which is non-invasive. Okay. Unlike optical methods which require very difficult or complex instrumentation for spectroscopy and all that, this requires a very simple approach which we call as reverse iontophoresis. Reverse iontophoresis. So in reverse iontophoresis, we apply an electric field, plus and minus, and this electric field actually creates a flow of ions, sodium and chloride ions. So whatever ions that are present inside the interstitial fluid, they start moving towards the electrode because of the electric field. Now when they start moving towards the electrode, what happens here is you can actually, what people have demonstrated is, you can also monitor non-charged or neutral molecules like glucose. It doesn't have any charge on it, but when the ions are moving, they are actually hitting the glucose molecules, and it causes the glucose molecule to come out of the skin, and then you can monitor them. Okay, so this was the concept that was present in Gluco watch. There was a device called Gluco watch which was commercialized, and then gradually it was released. It was pulled back again. It was not used because of many issues. You can look into that. So one of the issues was it had a very high MARD, mean absolute relative difference, between the blood glucose and the reading that it was giving. Then there were some reactions, if you continuously extract current, so there are some reactions with the skin, so irritation might be there. So that's why it was withdrawn. However, this is an interesting technique where you can have small devices, unlike like Professor Prasoon was saying earlier, optical device was very large, using the analogy of a bottle. So this, using a reverse iontophoresis, you can have a very small device, as well as if you play around with the way you are extracting the fluid, then you might be able to overcome certain disadvantages that are generally associated with reverse iontophoresis. Anyway, so here this is, if you look at the fourth picture here, you see this actually shows the capillary endothelial cells, and it tells us that molecules which are of very small size, they easily leak out of the capillaries and in the capillary blood flows. So they leak out into the interstitial fluid. So these are the molecules that we can easily monitor: ions, glucose, lactate, cortisol, levodopa, insulin, which is slightly larger, antibodies, even antibodies also. And then you have the inflammatory biomarkers, which are large in size, but still under certain circumstances you can also monitor them. So now coming to the next phase, once you have the so we know that we are actually monitoring from the interstitial fluid, or using reverse iontophoresis to extract the interstitial fluid. So now actually what are the analytes that we are interested in, and what electrochemical method we can use. So electrochemical methods are cheap to implement. They are not very expensive to implement. That's why, and you can also compress them. You can make it into a small chip, electronic chip, and you can easily actually miniaturize them. So that's why you have the, so what are the electrochemical sensing modalities? For sodium, generally we use potentiometry. So all sorts of, we can use ion selective electrodes, ISE, which has an ionophore layer on the electrode, and they allows only selectively certain ions to pass through and they block the other ions. So these are ISEs. pH also you can monitor. So we'll look at pH today and also in the live demo. So glucose, these actually use a chronoamperometry, lactate also, and then ethanol, which is alcohol actually. So they use various enzymatic methods, which we have already covered in detail. Enzymatic methods, and you can also have non-enzymatic methods also, but for stability these enzymatic methods are quite stable and repeatable, so that's why people tend to use them more. And then you have other markers such as uric acid cyclic voltammetry, then square wave stripping voltammetry, and all these things. So if you want to do, let's say, a label free detection, then you can also use EIS, electrochemical impedance spectroscopy, where if when the target analyte sticks on the surface, the impedance of the electrode changes, and you can use that as a measure of concentration depending upon the impedance. So you can actually, so this table actually summarizes what is my analyte and what is the method to detect it. So it's a nice link between them. So let's say you want to implement a sodium sensor, then you would go for potentiometry, unless you want to develop your own method, of course. But the traditional method is potentiometry. So similarly for glucose, generally people use chronoamperometry. So in this way, there is a link between the analytes that I want to monitor and what is the sensing modality. Okay, now as engineers and scientists, we deal with complex problems. So in order to solve a complex problem, what we try to do is we try to break down that complex problem in very simple steps. So that is what we are showing here. So it's like a block diagram, or step-by-step process where we can go from development to deployment. Okay, so what are the steps? First is the design phase. So here you design your sensor, you look at the material properties, you look at the transducing mechanisms. So if you are doing some sort of maybe let's say you want to implement flexible sensors, what should be the material properties, what is the Young's modulus that you require for your material, and all these things come into the design phase, where you actually focus on sensor design. So here you might, we require the electrochemistry experience, and we also require material scientists. So all these different kind of, experience are required at the design phase. And then after we have designed the sensor, we look at the sensitivity, selectivity, stability. So today, Professor Sanjiv in the morning, he showed you different, you can see parameters which are actually required to validate a sensor, how good your sensor is. So these are the parameters: sensitivity, selectivity, stability. Then we have MARD, mean absolute relative difference. So this we will look in more in detail at later lectures, where we are using AI to actually improve the MARD. And then we have the dynamic response. Dynamic response means how fast a sensor responds. Let's say in the body, the analyte, let's say glucose is changing. Let's say within 1 minute it is going from X to Y. Whether your sensor is responding as fast or not. So that is the dynamic response. So that is, so you are not only, because you are doing it for continuous monitoring, you are not only doing spot measurements, you also want to do the dynamic response. So if let's say I am moving from X to Y milligrams per deciliter, so whether your sensor is responding at the same rate or whether it is slow to respond, that is what we call as dynamic response. Okay. Now, once the design phase is complete, we move to manufacture. So this is where actually this design phase is very important, because all the subsequent steps, they are actually in a way implemented in the design phase. So whenever we are designing a sensor, if we want to take it to market, then we want to have a process which is scalable. So ideally we want to, when we take it to factories to manufacture, they already have their own manufacturing processes. The more similar to that manufacturing process, the better it is for us. Okay, so all these considerations, like in addition to sensor properties, we want to make the device scalable. Scalable means we can go from small volume to very large volumes easily. So that is what is being considered at the manufacturing phase. And here we do device to device variation. We check if we manufacture, let's say 100 devices, what is the variation that you see across devices. Ideally we want to minimize the variation. So this is in the manufacturing phase. And then we have large scale testing. So once we have manufactured the device, we want to actually validate it. So this is where you might have clinical trials, large clinical trials. Okay. And then clinical trials have different phases, the safety, phase 0, phase 1, phase 2, phase 3, like this, many different phases are there. So all of this comes under validation. And once we are happy with the validation, the final stage is deployment. Okay. So deployment is another, you can say it's an unknown step. We don't know whether once it goes out into the field, when we are using it in the lab, we know, okay, we should use it in a such and such manner. So now you can provide this advice. Let's say you ask the user to wear it properly on the, so you ask the user to implant it on the, let's say on the back of the arm. Maybe the user is not very attentive, so they might implant it somewhere else, and then the sensor is not working. So there are many factors which and unknown aspects which are there in deployment. So once the manufacturers, they actually take feedback from the users, and then they, if required, they go back to the drawing board, and if necessary, they actually release another version of the sensor. So all of this, and when doing this another second version of the sensor, they might improve the sensing layer ability or sensor capability, or they might also want to change the instrumentation that goes inside the sensor, depending upon user feedback. But broadly, these are the 4 different steps: design, manufacture, validation, deployment. Right, since we are all working in sensors, let's see what is the roadmap, where the industry is heading, where we are going. So what is the next step? So here we are in sensors 3.0. So the next step is sensors 4.0. Okay. So what we want is, we want AI on sensors. So nowadays, most of the AI is present in the cloud. So you have the cloud where sensor data is going, and then you are using AI models to process, train, and then deploying it back to the mobile or edge device. But ideally, what we want, for data security reasons, the AI to be present on the edge, on the device itself. So this is the next step, where we call it as connected intelligence or sensors 4.0. And that is where, we want to develop AI models that can be squeezed in small platforms, right. So that's what we call as, once you squeeze AI on the sensor, we call it as intelligent biosensors. Simple thing. Okay. So like I told you, as engineers, we try to break it down into different steps. So we have actually developed a technology stack. So what are the different technology requirements in an intelligent biosensor? So the first stack is this one. So it's just an easy way to understand. The first stack is the microneedle fabrication. Okay. So that comes under the first technology stack or layer. Okay. So then we have already looked at various fabrication methods. Professor Sanjiv has already developed and explained to you in previous lectures how to develop microneedles. Then we have, so we have the microneedle array, and then we have the sensing layer that also comes in the first stack, where many different methods are there. You can use an enzymatic method, you can use plastic antibody, which is quite interesting, which we learned earlier. So many different sensing methods are there, and this sensing method or layer is dependent on the analyte that you are actually trying to monitor. Okay. So all this comes under stack 1. And then as we insert the sensor into the body reacts. So we need an anti-fouling layer. So there are many different anti-fouling layers that are present that can be used on top of your sensing layer. These anti-fouling layers have to be porous as well, so that they allow the analytes to pass through. And then on top of the second stack is actually the instrumentation. So this is what we are actually looking at today, right now, in the current session, which is the instrumentation. So this is the current session that we are looking at. So in the instrumentation, we will have amplifiers. So if you don't understand amplifiers, there is no need to panic or be scared. They are very simple and easy to understand, and I hope by the end of the session you will be able to understand it properly. So amplifiers, you might have analog filters, you might have filters, and then you might have something called as ADCs, analog to digital converters. Okay, analog to digital converters. Now once your sensor is, so this will actually convert your current that is coming out of the sensor or voltage that is coming out of the sensor into a digital series of bits. Okay, so that is the language that is understood by computers. So we all talk in analog, but the computer understands in digital. So even this microphone has a digitized an ADC which converts voice, which is analog, into digital. Similarly, sensor signals are all analog, current, voltage, continuous signals, they are digitized into digital bits. So once it is digitized, the next step is to, we can take that data to the cloud or to the mobile phone, wherever we want. So that's why we need some sort of a communication protocol. You can have Bluetooth. So that is your second stack, the instrumentation. So the first stack is the sensor, then you have the instrumentation, and then since we want a connected system, we have the third stack, which consists of the decision making algorithm. And this decision making algorithm, there are many different ways to do the decision making. You can use control based algorithm, or you can also use AI nowadays. And then whatever decision, whatever output you require, I'll give you some examples. So that decision is actually can be given to an actuator. So actuator is like an insulin pump. So insulin pump is an actuator. So let's say I'm monitoring glucose. The sensor is collecting my glucose data, processing it, and let's say the AI says that, okay, I need to inject glucose. Then this information goes to the actuator or the insulin pump, and then this is again given back to the body. So if you focus here, so there is a closed loop system here. You see from the body, so it goes like this, and then it comes back. Okay, so you are the start of the loop is the glucose level sensor, and then you have the decision making, and then insulin which again affects the glucose. So it's a closed loop system. Okay, so that is what we call as a closed loop system. So that is the third stack, and the fourth stack is data storage. You can store it in the cloud. You can actually do the AI on the cloud as well. It's up to you, depending upon what type of AI you're using and what type of hardware you're having. So this is the fourth stack. Okay, so these are stacks. So in an intelligent biosensor, you would have 4 different technology stack, and this stack is meant to simplify things, help you understand and identify which area you can contribute. So let's say my expertise is more on the electronics and AI side, then I would like to work in let's say stack 2 or stack 3. So I would need help from stack 1. So I can collaborate with someone who has more knowledge in stack 1. Okay, so this is a way to simplify methods. So this is a typical closed loop system. We call it as artificial pancreas, where you have it starts with the sensing. So this is my first step. Then the control algorithm, which controls the actuator, and then again it goes back to the glucose level. So that is how, and this is my CGM sensor. So these are the old CGM sensors, which are actually Empatica CGM sensors. They used to be on the waist, and nowadays CGM sensors are located on the arm. And this is the insulin pump. This is the insulin pump. So the insulin pump is still being used like this, but the CGM sensor has moved from the waist to the arm, maybe due to tolerability and comfort reasons. People have moved it. And also one more thing to note here is there are certain devices in the market which actually combine sensing plus actuating. So you have both the sensor and the actuator in the same single device. So you will be wearing a single device, small device on the arm, which will have the sensor as well as the actuator. So there are certain devices also like that. One more thing to note here is one of the reasons why people keep it separate is, insulin has limited supply. So people need to replace the insulin cartridge, you can say, or the injection. So once it finishes, they replace it new. So when you have it in a single device, I don't know, I'm not sure how it works out. So this is the solution that we have generally. So that's an insulin pump, very small. It's like a credit card, credit card or debit card size. And then you have the sensor here. So this sensor actually, it goes, it's a long one. It's actually subdermal, right? Yeah, subcutaneous. Sorry, subcutaneous. So it's not subdermal. So it's subcutaneous. Subcutaneous means it penetrates the dermis and goes deep into the skin. That's why you see this is the sensing needle. This is not actually a needle. It's a flexible polymer substrate. So they use a device to puncture the skin insert the sensor and take it out. We will show it in the lab also in the during the hands-on session. So this is the current solution. So this is a much more better way to understand how the interstitial fluid works. So you have the this is the layer of the skin. You have the epidermis, dermis, and the subcutaneous layer. So this is where the CGM sensor would go up to the subcutaneous layer. Microneedles go up to here. So these are the microneedles. Okay, and then because when you go deep into the subcutaneous layer, you might encounter some blood capillaries, and when you are actually removing the sensor, sometimes you might have some blood which might come out because they are actually subcutaneous. So, and this is actually showing the diffusion from capillary to the interstitial fluid, the fluid flow, and these are so the diffusion actually is dependent on the size of the analyte, small, medium, large size. There are different sizes of the analytes which are actually you can detect. So up to 7 kilodalton you can easily detect. Is it clear? Right. So a much more detailed way. So the much more detailed diagram here. So you can see here these are the analyte sizes. So here we have free paracellular diffusion for small molecules like ions, glucose, urea, cortisol, vancomycin. Sorry, ions, glucose, urea, vancomycin, cortisol. If you want to monitor, let's say, insulin, so this comes in under medium solute. So this is actually something we call it as transitioning. So moving from paracellular diffusion, and as the size of the analyte increases, the diffusion actually becomes harder and harder and harder. Okay. So during inflammation, if the epithelial endothelial layers are actually become larger, the gaps between them, so they can actually diffuse easier, but that is only during inflammation, some sort of inflammation. So if that happens, you can actually detect this is an inflammatory marker, IgM, IgM. So it's around 990 kilodalton, so very large size molecule. Right. So one more thing to keep in mind, since we are working on the engineering side also. So when there is diffusion from the capillary into the interstitial fluid, it's actually, you can actually model them as a first order differential equation. And when you do that, there is a delay. So there is actually a delay, physiological delay that happens. So there is a delay if blood actually, let's say at 4 p.m. your glucose is spiking in the blood. So this delay will cause the spike in glucose to be detected around 10 minutes later. So there is a delay. So there is a physiological delay. So that is why we use AI to forecast the glucose level in advance, so that there is no problem with dosing and other things. We use some data processing methods to forecast the delay in advance. So these data processing methods we will look at a later lecture. But that is why we need some sort of a data processing approach to account for this delay. And this delay happens because of diffusion dynamics between the capillary and the interstitial fluid. Right, gradually we are moving more and more towards the engineering side. So as I told you, it's about breaking down the complexity into simpler block diagrams, easier to understand. So we have the biosensing element here, okay. Then we have the transducer, and then the transducer through the, let's say we are doing potentiometry or amperometry, it will generate an electrical signal which we are going to detect. So the first method that we have seen up to now, so it is, let's say we are doing glucose detection. The signal to be measured is actually a current. Okay. So this is actually measured. So we have 3 electrodes: the working electrode, reference electrode, and counter electrode. So what we do is, in amperometry, we apply a fixed potential between the working and the reference electrode, and we measure the current that is flowing through the counter electrode. Okay. So when we apply this constant voltage, redox reaction happens, and whatever ion exchange takes place, that is actually detected through the counter electrode. Okay. So that's why we say current at fixed potential. Now in potentiometry, we do not apply any voltage. We simply measure the voltage between the working electrode and the reference electrode. Okay. There is no flow of current between working and reference electrode. Okay. So then we can measure we simply measure the voltage between the working electrode and the reference electrode. This is for potentiometry. So in voltammetry, what we do is, generally it's used for heavy metal detection. So what we do is we do a potential sweep. So this voltage that we are keeping constant, between the working and the reference electrode. So it's no longer constant. You can use some sort of a sweep, so triangular waveform, which you can actually use to change the potential and then look at the current that is coming out, and you can do your analysis. And then there are some sensors which are impedimetric. So we look at change in impedance. So these are level free sensing, and generally surface binding happens in impedimetric sensor, which causes the sensor to change the surface impedance, and then we are able to detect it. Okay, so there are other types of sensors which we are not going to discuss, which is conductometric and capacitive sensors. Most of the touch screens, they are capacitive sensors. Okay, so far so good. Moving on, so I just wanted to give you a feel of how the glucose sensor looks like. We also have some samples. So these are the typical glucose sensors. We have the GUARDIAN sensor from Medtronic, Abbott Freestyle Libre sensor. This is the sensor which we have in our lab and which we will demonstrate also during our hands-on session. If you wish, you can also implant it. And then we have, if you wish, I want to say, there is no, we are going to implant ourselves, but you don't, it's up to you if you want to try out. So then we have the Dexcom G6 sensor. So these are the big players in the glucose sensing CGM market, so to say. And we have already looked at these things, so I don't need to go in detail. The first generation, second generation, and third generation sensors we have discussed in detail previously. So all of this actually use enzymatic sensing. So now coming to the artificial pancreas, so we will mostly be looking at the glucose sensor and how we can extract the glucose signal here, glucose concentration. That is our main focus of the lecture here today. Glucose concentration, how we can actually, what kind of instrumentation we require to measure the glucose concentration. So that is what we will be mostly focusing on. So overall, I just wanted to give you a feel of how the entire system looks like. So the entire system, this is a way to model the entire closed loop system where you have the controller. This is the, you can say this is the algorithm, the AI model or algorithm, which is like a controller. Okay, and then which controls the insulin pump, and the AI algorithm is dependent on this signal here. So this is the control objective. Let's say I want to maintain my glucose level within a certain range. So that is the objective, that is my range in which I need to control the glucose, and this is whatever signal is coming in. So it finds out the error, it calculates the error difference between the objective and the real term value, and then accordingly the controller will generate a signal U, which will actually control the insulin pump and try to bring it down within that same range. Okay, and this is how you can actually model an individual with type 1 diabetes. So there are many different in silico models of type 1 diabetes. So UVA Padova model is a very popular model, that in silico model, UVA Padova. So it is developed by University of Virginia and Padova University, which is in Italy. And this model is actually FDA approved. So let's say you want to develop some AI models and you want to try out some things without collecting actual data. Then if you show the UVA Padova model, if you use the UVA Padova model to generate some patient data, and you can test your AI quickly on that. Of course, the holy grail is actually the clinical data, but you can start with the in silico data without having to wait for the clinical data. Okay. So there are many different approaches. So this controller, we can use it as a PID control. That was the old way in which how people used to do. Then we have the bio-inspired modeling, beta cells. And recently people started using the deep learning approaches. So this deep learning approach, we will look more in detail in a later lecture that is going to come. So generally, okay, so now coming more and more towards the instrumentation side. So amperometry is used for glucose sensing. In amperometry, actually, as I have already told you, you maintain a constant voltage between the reference electrode and the working electrode. And then whatever current that is being generated in the reaction, we try to measure using the counter electrode. Okay, so this is the amplifier that we have here. So I'll show you some front ends that we can use. So you have the reference electrode. So this voltage actually appears here. These are so this will again be VDD by 2, and this is the counter electrode through which you are actually measuring the current. So if you look at the voltage between working electrode and reference electrode is actually 0.65 volt. Okay, the difference, voltage difference. So this could be the oxidation potential that you require to start the redox reaction, and then you measure the current that flows through the counter electrode. Now in the olden days, previously, let's say we did not have any electrochemical sensing front end. So let's say I want to if you think of an engineering project, let's say during your BTech days, early BTech, first year, second year, you might be doing a project where, let's say, there is a sensor which will detect, let's say, movement and turn on the light. So one of the components that is required is a microcontroller. So the microcontroller will detect the movement and then it will turn on the light, right? So we did not have this kind of application or this kind of, let's say, device in electrochemistry. Okay, so if you want to develop a sensor, you have to have a potentiostat that you have to buy, which is a big device, and you have to use it. But in recently, around, I would say, let's say 2022, 23, around that time, analog devices, they have released this chip. So this AD5940 or AD5941. So this chip actually, if you can bring that. So this chip actually you can use to do you can make your own potentiostat. So you don't have to actually if you wish, you can also make a much more smaller potentiostat. So this is a evaluation board of AD5940. It has a chip, AD5940 chip, and this actually has the functionality of a potentiostat. Okay. So you can actually make your own, if you want, you can make your own potentiostat using this chip. So you don't have to worry about how will I implement amperometry, how will I implement potentiometry. All the functions are already there in this chip, AD5940 or AD5941. Okay, and it has a very comprehensive and detailed data sheet that you can follow, and there are many examples on their website as well. So you don't have to program it yourself. You can use these examples, and then if you want to modify according to your need, you can modify the program. Okay, so we'll look into this a little bit more in detail later on. And this chip size is very small. So this is in millimeters, as you can see. So the size is, you can say, 3x4, very small, 3x4 millimeter. It's very small, 3.6x4.6 or 4.2. So it's a very small form factor chip that we have. And all you need to do is you need to design your own PCB. And there are many more tutorials that are available to help you design your own PCB. But once you do it, you can easily have your own potentiostat. You don't have to rely. Let's say I am developing a wearable device where I need to develop a potentiostat, or where I need to use a potentiostat for any marker. I don't have to have a bulky potentiostat that is present in the lab. I can design a small one using AD5940. And this is called as an evaluation board. I'll talk about it. But the chip itself is very small. So you can have your own PCB, much more smaller form factor, and you can have your own CGM sensor using this front end. So this front end, analog front end, was not present previously a few years back, and we had to design our own front ends, own amplifiers and everything. So I'll give you a flavor of the old approach and also the new approach using this one. So this is a chip which one of my previous colleague, she actually made, Sara. So you can see here there are many different components here. The reason I wanted to show you is there's some few things which I wanted to actually explain. So this is your potentiostat, and then you have the ADC. Okay, so this is the analog to digital converter. SPI stands for serial peripheral interface. So this is another interface that you can use to extract digital bits serially out of the hardware, out of the, let's say you have this AD5940. If you want to extract some digital bits out of it, then you can use the SPI protocol. Okay, so we all are familiar with USB. So USB is one of the communication protocols. Similarly, SPI is another communication protocol. So both of them are serial. Okay, so USB stands for Universal Serial Bus. This is again a serial protocol, SPI, serial peripheral interface. Okay, so using this you can actually extract data out of it. But previously this design used to be done entirely customized. So this is the counter electrode, and the red arrow shows the direction of current. It flows inside the potentiostat, going inside this triangle here. and this triangle has some electronics internally, some transistors which measures the current, and then you measure what is your concentration. So I'll show you an example of this. This is a much more detailed diagram of the previous chip. So the chip actually, this is called as an ASIC, application specific integrated circuit, ASIC, application specific integrated circuit, integrated. This is called as an ASIC. Let's say I have an application for sensing, then I would design an ASIC. Now this ASIC design process is a very tedious and long process. Okay, so at least it takes 1 year to design a chip, have it fabricated, bring it back to your lab, test it. Okay, so it's a long process. So generally in wearable medical devices, we have two trade-offs. Trade-off means we have to think of majorly two aspects that we need to look at. One is called as power, and the other one is size. Okay, so if your device is consuming a lot of power, that means you need to have a big battery on it, a large battery. So then your device size increases. So if you are actually increasing the power, device size will also increase. Ideally we want something which is low power and small device size. So this is where actually you can do some intelligent or many clever techniques that you can apply in your ASIC to ensure that your device is small size and low power consumption. Okay, so that is where actually so that requirement, previously the requirement for ASIC was there in electrochemistry because there was no available platform. So nowadays we have a platform which is AD5941 or AD5940. So we do not require an ASIC per se. Okay, only if I want to, let's say this chip has a certain power consumption and I want to have lower power consumption, then ASIC design is required. If I'm not too much worried about the power consumption or the if I don't have any justification for going smaller chip size, then I can use AD5940. It's easy to program, easy to use. I can buy off the shelf. I don't have to actually worry about these things. Okay, so this is a much more detailed block diagram of the AD5940, where you have the current channels. So current channels, what type of electrochemical method uses current, generates current? Amperometry. Very good. So it is actually amperometry. Okay, so these current channels, if we look in much more in detail, so they have something called as a TIA amplifier, trans impedance amplifier, which will convert amperometry into voltage, and this voltage is actually digitized using ADCs. So using ADCs, these ones, sorry, these ADCs actually digitize the output from the current channels. Voltage, potentiometry. Okay, potentiometry. And then we also have impedance, for impedance measurement, EIS. So it's a nice chip with lot of different, almost all functionalities, modalities packaged within a small chip. So it's good to remember this chip, AD5940 or AD5941. So if you are developing wearable sensors and you don't want to do ASIC design, or you don't have the, so this ASIC design requires in-depth knowledge of electronics. So if you don't want to do that, then you can go for AD5940, AD5941. And this is the evaluation board of the chip. So generally what happens is, let's say I want to test, this requires some programming. Okay, so if I want to program the chip and test it and use it without having to develop my own PCB, then I can use this chip, this AD5941 or AD5940 evaluation board. These are my electrodes that are required, reference electrode, working electrode, all the electrodes that are needed for electrochemical sensing. I can insert it, I can program it, I can test it. Okay, once I'm happy with the program, once I'm happy with the application, then I can design my own PCB with the same chip. So I just need to transfer the program into the chip. So I don't have to do the programming again. So in that way, so that is the typical design phase. That is where you use the evaluation board to test the whole process. A much more in-depth diagram. I don't think we require to go into all these things, all the details. If you wish, we can discuss it at a later stage. But this is the AD5940, fantastic chip, very useful. So this is the block that we generally use for amperometry, for sensing the current let's say out of a glucose sensor or a lactate sensor. Clear? Okay, I just wanted to highlight also. So this is actually, I just wanted to show you how it has been used in a platform. So this is a paper recently published by the same group, I think Joe Wang, where we have multi-analyte sensing. So multiple sensors are implemented. So they also have a microneedle based platform, which is this one. It has a microneedle sensor patch. So I think they had glucose, and then you have lactate, and then alcohol. So these are few analytes they were trying to monitor at the same time using the same platform. So within the same microneedle platform, they have these multiple sensors that are implemented, and they use this AD5941 or successfully, and they have demonstrated this. So this was published in Nature Biomedical Engineering, and yeah. So this device is detachable. So the electronics itself you don't need to throw. So let's say you are using a sensor. You can use the sensor for a limited amount of time. You cannot use it infinitely or for a very long amount of time. So you can use it, say, let's say for 1 week or 2 weeks, and then you have to throw away the sensor. So instead of throwing away the electronics, you can simply detach the array, throw it away, and replace it with a new array. So that's what they have actually shown here. And so here we have the this is the transimpedance amplifier that I was telling you about earlier, which converts the current which is coming from the working electrode. So they have multiple working electrodes, working electrode 1, 2, 3, 4 different analytes, but they are sharing the same reference and counter electrode. So depending upon, let's say I want to monitor the glucose now, then I will switch to a certain working electrode. If I want to monitor lactate, then I will switch on another electrode. So like this, I can selectively monitor certain analytes while at the same time using the same counter and reference electrode. And this goes into a TIA, transimpedance amplifier. So the input here is current, output here is voltage, and then this voltage, this is called as a multiplexer. But anyway, this voltage is again amplified. Amplified means the value is increased. So PGA stands for programmable gain amplifier. So let's say I'm having a amplifier. So let's say I'm having a signal which is 10 microvolts. I want to amplify it to 100 microvolts. Then I will multiply it by 10. So the gain is 10. I want to multiply it by 1000. So 10 microvolt becomes 10 millivolts. So this is what I can choose, how much gain I want on the signal, and then or the multiplying factor, and then this voltage. So this PGA or programmable gain is required to fit within the range of the ADC. The ADC has some limitations. So it can only digitize signals between, let's say, X microvolt or X millivolt to Y millivolt. So in order to fit within this range, I have to amplify the signal. And once I amplify the signal and fit it within the range of the ADC, I can now have the analog signal converted into the digital signal, which can be understood by computers, which can be processed by computers. Okay. So if I want also, I can implement digital filters. I can have low pass filter. So let's say when we are moving fast, when we are running, so it causes, the sensor signal causes some spikes. So we can remove these spikes using the digital filters. And let's say we are sitting near electronic devices. There is some feed through or electromagnetic induction. 50 hertz we have AC signal which is powering up our devices. So that 50 hertz might also come in. So that can be removed using filters. Clear? Okay, moving on. So we have seen how actually, so glucose and pH, these are very common biosensors that are present in labs in and around. So very common markers that we generally monitor. So I have shown you the instrumentation that is required for glucose sensing. So let's have a look at the pH sensor. So both this glucose sensor, the enzymatic glucose sensor and the pH sensor, you will see more in detail during the hands-on session. So you yourself can implement a glucose sensor. You yourself can actually implement a pH sensor as well. So this, I wanted to show you some work related to the pH sensor, and this is the last part. And after that, we'll take questions. So in the pH sensors, generally this is the gold standard, the glass electrode. So that is the gold standard in pH sensing. And then we can have micro spikes or microneedles. Then we have something called as ISFET. Does anybody know what is ISFET? Ion sensitive field effect transistors. So this graph actually is a very rough graph, but it is a qualitative graph. It tells us that the SFETs are very small in size, in dimension, but they are not invasive. Okay, you cannot insert it into the body as such. And then you have the glass electrode, which is very large in size, but still you cannot insert it into the body, you cannot carry it with you. So that's why we have the microneedles or microspikes or subdermal sensors, which can actually be penetrated and worn for long-term monitoring, continuous monitoring. So these SFETs and glass electrodes, we cannot use it for long-term continuous monitoring, but microspikes we can. So in this one, we actually use iridium oxide microelectrodes. So these iridium oxide pH electrodes are very, very good pH electrodes. So, and easy to implement as well. So we'll tell you the method how to implement it in the lab. So this is actually a gold coated microneedle sensor array, which we have functionalized it with iridium oxide. So this is the sensing mechanism. I think Sanjiv will be better able to explain this. This is a standard iridium oxide, iridium 3 to iridium 4. So this iridium 4. So there is a transition between different oxides of iridium 4 to iridium 3, iridium 3 to iridium 4. So that is a redox. So it's a reversible reaction, and the sensitivity is around 55 to 75 millivolt per pH. And this follows, as you all know, Nernst equation. It follows the Nernst equation, and this generally, when this 75 millivolt is what we call as a super Nernstian response, a very highly sensitive device that we can have. And we have very small dimension also. The length is 100 micron of the spike, the microspike or microneedle, whatever you call it. Good response time. It changes very fast, 5 seconds. So if your pH is changing, although pH in body doesn't change that much, but this is the kind of response time that you have in pH. So we'll show you this, implement the iridium oxide sensor as well in the lab, and you can see it on the hands-on session. You can get a feel of it. So what is required? So this is some work which I did in 2017 when there was no AD5940 available. So in today's scenario, I would go for AD5940, AD5941. I can immediately or directly interface the pH electrode with the platform sensing with AD5941 or AD5940. But what we need to keep in mind in pH, actually this is very important. It should have low input bias current. So what happens here is, generally when we make amplifiers, these amplifiers actually, the ideal input impedance, let's call it IZ, or let's call it ZI, let's say, input impedance is infinite. So infinite means there is no current which is flowing into the terminals. Okay, so ideally we have infinite input impedance, but let's mark the word ideal. So there is always some small current which is flowing inside. Okay, so that we call it as I bias or bias current. Okay, I bias. So the input bias current should be as small as possible. There is a reason for that. I'll explain as well. Very easy to understand. So when you are designing anything for like pH sensing, the input bias current should be in femtoamps. So in AD5940, if you go back to the data sheet and you can check what is the input impedance of this device and what is the input bias current that is expected for the voltage channels. Okay, so if it is in femtoamps, then it's good to use for pH. So even in picoamps also you might be able to escape, but I would say that go for femtoamps. So what happens here is, the reference electrode, the silver chloride reference electrode. So generally what we have here is, let's say this is the pH electrode. This is a very simple diagram. I'm not doing the detailed. So this here you would have the reference electrode, which is AgCl, and if you have seen the AgCl reference electrode, it is a glass electrode. Okay, and there is a salt fit at the bottom. Okay, so this has very high impedance. So this AgCl electrode has very high impedance. Okay, so if there is a small current, even a small current that flows through the electrode, because of the high impedance, it gets multiplied, and you have a voltage signal, which may not be due to pH change. It may be due to this input bias current which is flowing. So what we will have here is, generally in pH sensors, I will show you in later diagram, here we have voltage, and let's say we have time. So this is known as the OCP, open circuit potential. So the potential that we are measuring between the reference electrode and the working electrode, we call it as OCP, open circuit potential. Ideally, the OCP, if you are inserting it in a buffer, let's say the pH is not changing, ideally the OCP should be like a straight line. But due to this bias current, input bias current, there is a drift. There is a small drift. So if you have very high input bias current, even in microamps or picoamps, there will be drift will be more. So femtoamps means little bit more stable. So you will have less drift. And there are some problems which arise due to drift. You can use signal processing methods or even AI driven methods also that we will see in a later lecture. You can remove these non-specific changes or variations, and you can see the nice pH signal, pH change in vivo. We will see at a later stage. So ideally you want to have very low input bias current. Why? Because I want to have low OCP drift. And the reason of having low OCP drift is that the measurement will be more accurate and stable, easier to measure. Okay, so here this is an amplifier LMP7721. So you can use this amplifier LMP7721. So again, this is, you can buy off the shelf, you can buy it online, you can implement it yourself. There are many reference designs present, you can directly use them. So it's easy to design, easy to use. Okay, so this just a brief, wanted to show you some results also about the iridium oxide sensor. So this is a micro spike that we developed, 3D printed model. First we have a 3D printed model, and this one we can also have injection. So better is to go for injection molding. So I think we used injection molding actually. So injection molding, you can have very small micro spikes like this, as you can see in the picture here, and then you put a polymer mask on top of it to make these lines or conductive contacts, and then you cover it with another polymer layer so that it doesn't interfere with the measurements, like Sanjiv was saying earlier. So you have only the spikes which are exposed, and then you have the tracks there to connect, to extract the signal. And then, so these are the different steps that are required to make the pH sensitive. So once you have made these electrodes, once you have sputtered it with gold, because a polymer is non-conductive, you can actually gold sputtering also gives a uniform layer, and then you can functionalize the gold layer with iridium oxide, and then you can calibrate it. So this is the process of oxidation, or you can say how you can electroplate it, or you can I don't know if this is the right terminology. So generally we use cyclic voltammetry to create a layer of iridium oxide on top of the microneedles. And then this is the nice curve that we have been seeing. This is the CV curve that you would see for functionalizing the iridium oxide. So which tells us that the nice the layer of iridium oxide has formed on top of it. So and then of course you can calibrate it. You can use different pH buffers to calibrate your sensor and see if your sensor has a linear response. If it is linear, what is the range, linear range, what is the sensitivity? So sensitivity is around 0.075 millivolts per pH. Okay, so for every pH change, 1 unit of pH change, your potential will change by 0.075. Sorry, this is volts. This is volts. Or in other words, you can say this is actually equal to 75 millivolts per pH. Okay, so for every unit pH change, you would have 75 millivolt change in voltage. Okay. Okay, and linearity is important for the sensor, so that you can have a constant sensitivity. If the sensor is not linear, then your sensitivity will change and it's hard to estimate. Okay. Okay, now this is actually at that time there was some experiment which was going on on hearts, excised hearts, ex vivo heart. So where we wanted to see if using these pH sensors we are able to check. Let's say if there is ischemia, that means oxygen supply is restricted, pH will change in the heart or not. So this is a male Vista rat heart which has been excised and the microneedles have been implanted on it. And this is the kind of histopathology after you have implanted and you have taken out the microneedle. You want to see what is the damage on the tissue that has been done, or what is whether it has really penetrated or not. So these are some imaging. So overall, so we tried to validate these microneedles using in this application where you induce heart attack or ischemia, and then you try to revive the heart again, so and see if there is any change in pH or not. So this shows us, this result here shows us that, you know, if you look at the pre-impalement ECG and the post-impalement, there is not much of a difference between pre and post. That means the heart is not damaged because of implantation. Okay, and this is where we are inducing the ischemia or heart attack, you can say, by cutting off the oxygen. So this is the kind of drift that I told you about earlier, and this actually had this LMP amplifier that I had shown you. There is still a little bit of drift present in this in this recording. So this is the OCP drift. This is the OCP drift. It happens due to very small current that flows into the amplifiers, pH amplifiers. It can be femtoamps, but you see that there is a drift. So what you do is, if you look at the dotted line, you can estimate the drift, linear drift, and then subtract it from the signal, so you get a flat response. That's how we do it, and that's how you can actually remove the OCP drift, and then you can see the nice peaks in the signal. I'll show you in the next slide. Is there? So yeah, so these are, so we had around, if I'm not, so we had around 7 microneedles on the array, and you can individually monitor them using these, you know, so here you are actually inducing the ischemia, pH goes down, reperfusion, pH increases, then goes down, increases. In this way, it tells us that you can reliably monitor the pH in tissues. So that was the objective of the study. And then you see that there is little bit of drift also. Even after drift removal, there is slight drift is still present. So it's very hard to remove the OCP drift. So the absolute value, if you are calculating the absolute value, you have to be very careful about the drift. And this we combined it in a nice package PCB. So this is a flexible PCB cable, which you may have seen earlier, FPC cable or flexible PCB cable. It's connected to the electronics, battery, and everything is there. So now, just towards the end, we are almost going towards the end. So here, one of the challenges in continuous monitoring is the degradation in sensitivity due to biofouling. Once you implant the sensor, you see, so this is actually this, if you look at the right hand side plots, this is a CGM sensor. You know, initially it's a very vague, I mean, it's only one-off experiment, but it gives you an idea, a qualitative idea. So you see that the blood glucose, the finger stick reading and the CGM reading are more or less matching, but as you move on, there is a gap between the capillary blood that you are drawing from the finger prick and the measurement that is done in the CGM sensor. So this is with increasing number of days. So that means there is some degradation on the sensor, which causes the sensor value to vary from the blood glucose value. And then of course this is the linearity plot. So ideally they should overlap, but they are diverging as you go, you know, day wise. So this, you know, this variation is still acceptable within the FDA approved guidelines. So that's why these sensors are still being used up to 14 days. They are still within the range, but as an engineer, we try to think, can we solve this? Can we close this gap? So yeah, there were some sensors which were present in the market where they actually ask the user to calibrate the sensor daily, twice. So every 12 hours, you can take a finger prick reading, and they have an app where you can enter this finger prick reading, and whatever reading they are getting from the CGM, they try to close the gap. They try to, so that is a 2-point calibration that they try to do it. But of course it is inconvenient. If you are using a sensor, then what is the point of taking the blood glucose reading again? So ideally, so what people nowadays have, and we'll show you some sensors also in the lab. So they call it factory calibrated sensors. They have already calibrated it, and they know how much the sensor degradation will happen, whether it is within the range, FDA approved range or not. So, but ideally what we want is, we want these readings to be as close as possible to each other. Okay. So with that application in mind, we actually tried to see if we can do it. So this is one of our publications from our group, where actually we tried to probe the surface of the sensor using electrochemical impedance spectroscopy, and then whatever parameters you are getting from the EIS, you prepare a data set and link it to the sensitivity, and then you can actually attempt some sort of a prediction with respect to days. So you can actually say that, okay, at day 11 or day 10, what would be the sensitivity based on the EIS measurement of the sensor. So this is a very early proof of concept study that we did, which demonstrated that yes, there is possibility. Of course, you can expand it by going more in depth, try to do in vivo results and all these things, which we are trying. This is another attempt. So that was the glucose sensor, enzymatic glucose sensor. This is pH sensor. Here we tried to coat it with PVA coating, which is an biofouling resistant coating, and then you look at the impedance, you probe the impedance and try to find out. So we found out that, okay, with PVA coated, whether it is PVA coated or PVA not coated, there is some link between the sensitivity and the double layer capacitance. So when we have, if there is a deposition of biofoulants on the sensor surface, naturally your double layer capacitance will increase, and then there is a link with the sensitivity. So these kind of modeling relationships we tried to make so that we can actually predict the sensor sensitivity day wise without having to do the finger prick reading. So that's all, almost towards the end. So this is a platform that we have developed in the lab, low cost device, very easy to make, and we have integrated with the electronics. We can show you also later on. And this is the high density microneedle based platform, which Sanjiv is an expert in that. So we also tried to do some microneedle development, which we have made. But yeah, it's not perfect as yet. So this will give us, so you can say this is high density monitoring. So of the analytes. So this is kind of low density. So the more number of higher the density, better reading you will get, better surface area is there. So more accurate will be the reading. So that's all from my side. I mean, if you have any questions, you can ask. So overall, we covered the amperometry and potentiometry. What are the requirements from an instrumentation perspective, and what is the latest instrument that you can use, latest platform, which is AD5940, AD5941 that you can use. Yeah.
Sir, like you said, the sensor degrade over time. So which one should be easier for a person to develop it for increasing its shelf life, or to make it more cost effective, like in today's generation?
So I would say making it cost effective will be better, because what we got as user feedback, people who have actually, we did a small survey within people who are using CGM sensors. So they don't like to, they don't like the device being attached with them for very long amount of time. So it's more important that we, if the sensor is cheap, they can replace it frequently. So it's more of a comfort level. So people want to use it maximum for 1 week to 10 days. And they don't want it to be attached for a very long time. They feel a little bit uncomfortable sleeping and other things. So I would say, if you ask me, my personal opinion is we should aim for cost effectiveness, so that you can have more number of sensors replaced easily, rather than having one sensor for 1 month. So right now one of these sensors cost around 5,000, these CGM sensors. So if somebody is using it 2 times in a month, so the recommended use is 14 days. So every 28 days you would require 2 sensors. So that is 10,000 cost. Now imagine if you reduce the cost by 10 times. So let's say 500 or even 1,000, and you can have 10 sensors within the same budget, and it may be more comfortable, better to read. It will be more accurate because you are replacing it frequently, and there will be less engineering, stress on the sensor as such. The reason, all sensors are not same. Why we did this prediction sensor, sensitivity prediction, because you see, we have a glucose sensor. It may not react at the same way when you insert some other kind of sensor. So it depends on how, like what is the sensor surface that you are using. So the body might have a much more stringent reaction to sensors to different surfaces. Okay, so depending upon the it also depends on depends on the type of surface that you're using. Let's say the body reacts more quickly to a certain type of sensing layer, then you would require this sensor prediction within 7 days also. Okay, so from a glucose sensor perspective, my personal opinion is it will be good to focus on reducing the cost of the sensor so that people can use it more frequently, and reducing the weight of the sensor, make it more like a patch, like a band-aid. Many of us have used this band-aid. When you have a cut, we use a band-aid. Something of that weight will be really nice. You don't even feel it. So around we have sensors, so you can actually feel the sensor after the session. You can have a look what is the weight of the sensor and how it feels. You can have a look. Sir. Yeah. Yeah. Yes. Yeah. Yeah. Iridium oxide. Yeah. Yeah. So the sensing mechanism is different. So here the equilibrium reaction is different. So it is this iridium 3 to iridium 4. Just 1 minute. Let me just go back to that slide. So this is this reaction is different in a glass electrode. So this would be different in a glass electrode. The reaction sensing reaction is different. Yeah.
Okay, so the mechanism is totally different.
Mechanism is different. Yeah.
Totally different.
Yeah.
And is it like because of that actually you are getting this sensitivity somewhere, like because normal glass electrode we have read like somewhere 59, like theoretically like 59 millivolt per pH.
Yes. So it is because of this. Actually, I have shown you one interface reaction. So there are 3 possible interface reactions actually happening. So this is one of them. So that is why the sensitivity is varying from 55 to 75. So depending upon what type of reaction is happening, it will be different. So people have, so when you are coating iridium oxide layers on the sensor surface, there are many different protocols. It depends on the protocol also. So different protocols will give you different type of sensitivity. So in some protocols you will get which is close to glass electrode around 50, 55. And in some protocols you will have a super Nernst sensitivity which is around 70, 75. It depends on the protocol.
And then application point of view, like where you cannot use glass electrode, there you can use this.
Exactly, yeah. Where we cannot use glass electrode is the gold standard. So even if we make these electrodes, we always validate it with the glass electrode. So whether we are getting the same reading with the pH electrode that we have in the lab and the microspike that we have developed. So that comparison is always there. So if you make, let's say, a new array and you are trying to publish it, then you have to show the comparison with the glass electrode to convince the reviewers. So that is always there. The only application is, let's say, we cannot penetrate the glass electrode. There we use the iridium oxide electrodes for penetration.
Okay.
So
these glass electrodes are not in micro and semi-micro. I mean, no. Okay.
And I have seen some capillary type of structure glass electrode also there, but they are easy. They break very easily. So I remember using. We made some very thin capillary based glass electrodes for animal experiments, but they are not reliable. They break very easily. That is the when you penetrate. When you try to take it out, sometimes it breaks, the tip breaks and stays inside the body.
Okay. So another query regarding this chip you told is something like totally new for me at least.
So this one, right?
Completely.
Yeah.
Yeah. So because we have a big this potentiostat.
Yes.
My question here, can you use it for voltammetry?
Yes,
I mean,
yes.
you can change also the potential with
Yes,
time.
yeah, you can use it for voltammetry also. So they have some, they have functionality here. So this waveform generator, so this waveform generator actually you can use it for voltammetry also. So let's say you want a triangular waveform for
Uh-huh.
voltammetry, so you can program them.
Oh, okay.
Yeah.
And then
So-
the paper you showed that nature,
Yeah.
like three things it was like, analyzing.
Yes, yes.
Like it's like not in the same simultaneously. It
No,
was
not
measured?
simultaneously. No.
You have
So
to stop.
yeah, so they have this switch. So this is a switch here. So whenever they require a certain analyte, they will turn on that switch or select it. And because what happens is they are actually sharing the counter electrode and reference electrode.
Uh-huh.
So you
But
cannot
three
have
different
simultaneous.
working electrode you told, right?
Huh?
Three different
Three different working electrodes and one reference and one counter. So they cannot use all three working electrodes with the same reference and counter.
Okay.
So they are forced to use this. So if you want simultaneous measurement, you might have to have, separate.
Three reference,
Three
three.
reference, three, exactly.
Okay, maybe.
Yeah.
Thank you. And probably this LDO, like yesterday you
yes,
were
yes.
talking about,
So
today.
I will show you. It's there in this block diagram also.
Yeah.
Yeah. So here,
Maybe another one somewhere it was there. This is a total detail block diagram.
yeah. So you
Maybe in
see,
the-
sir, look at this
Yeah.
LDO. So it generates a constant voltage. Whenever you require constant voltage, so this will be operated. You can operate it with a USB, let's say, or a battery. Battery generally is 3.3 volts. So this will convert your 3.3 volts to a constant voltage LDO. So one output will be 1.82 volts. In that way, you can.
Thank you.
Sir, I have also a small question. In that continuous glucose monitors,
Yes.
The reduction potential was set at 650 millivolt.
Yes.
What if we go for the literatures,
Yeah.
the FAT moiety in the glucose oxidase or for even glucose also, they're very, they're close to zero. Like for glucose oxidase, it's 80 millivolt, and for glucose, it is 420.
Yeah.
Then why is it set for 650 millivolt?
So actually what happens is, you cannot use the enzymatic electrode GOX directly because there is problem of interference. So like we discussed last time, acetaminophen, it's an interferent, ascorbate, it's an interferent. So what people do is they use some layers. There is some layer called naphion, you must have heard. It's a polymer naphion. So they do these changes to the surface to enable to avoid the oxidation potential to be same as the interference. So that is the reason why you have different voltages. Okay, because of the changes in. So ideally, so once you make a sensor and you functionalize it with naphion or some other kind of polymer, it shifts, the potential shifts. So that is the reason.
Thank you so much, sir, for this informative presentation. And sir, I want to know that you have talked about the artificial intelligence and machine learning,
Yeah.
and you have just said about the accuracy and in comparing the things.
Yeah.
And as we are going towards the agentic AIs and generative AI and artificial general intelligence,
Yeah.
so sir, how these things will change the pace of the researches which is right now ongoing,
Yeah.
and how it will help in personal medications?
Okay, sure. So your question is a little bit broad, so I'll try to make it specific to this topic. Let's focus on generative AI. So I will show you in another lecture. What happens is when we use these CGM sensors, Sanjiv, you might know also. So you have also shown sometimes what happens is you might have missing data points, right? So if you are using a Dexcom sensor which has a Bluetooth device, let's say that Bluetooth is connected to my mobile phone. I have left the mobile phone over there somewhere out of range and I'm moving about. So there is a drop of data points are not present, they are absent. Okay, so you can use generative AI to generate the missing data points. So once you use that missing data points, then you are safe with your AI and the decision making. You cannot make any decision without data, so you require data. So if you want the data to be as close as possible to the glycemic profile of an individual, then you can use Gen AI. So you can use GANs. We have done that also to replace the missing data points and then use the decision. That is one answer to. Other than that, you can see there are many other applications of Gen AI. For example, many people have tried to develop an LLM for diabetes. Let's say somebody is having some issue with diabetes, or they want to ask a general query. So they can ask an LLM which has been trained on these applications, and LLM can, so like a chat GPT for diabetic patients. So it can answer specific queries. Let's say you want to ask what should I take insulin or not, or maybe what is my current glucose level, should I take a walk or not. Sometimes in type 1 patients, one of the concern is physical activity. So if they do very rigorous physical activity, then glucose level falls, then the insulin dose also changes. So if you cannot have the insulin dose which is same for without physical activity and with physical activity, because you might require less insulin to avoid hypoglycemia. So in that scenario, actually LLMs, which are part of generative AI, they can answer queries. You don't have to call your doctor and ask as such. At least for something quick query, you can ask. Then if you want to verify, you can ask a doctor. These are some applications of Gen AI in diabetes.
So I have one question. You talked about this instrumentation. So many a times during this electrochemical sensing, because of some other fluctuations happens, because this is oxidation reduction reaction, so may not be very smooth because of the diffusion problem or mass transfer limitation. So can this instrumentation can handle those fluctuations, or it need to be cured or
Yeah.
corrected with the algorithm only?
No, there are so here we have we have digital filters here.
Okay.
You can see there are digital filters. So these you can use some basic noise removal. You can use digital filters to do that. Let's say you have a sudden spike due to movement, or let's say you fell down or you moved your hand and there is a sudden spike. You can use a digital filter to remove that spike.
So instrument can
Yeah,
itself
instrument
take care
can
of
do it. You have to program it. So this is programmable. So it's like a microcontroller. A microcontroller generally has mostly an ADC, which is an analog component. Rest are digital. So this is a nice platform because it combines both analog and digital together. You can program it also if you want. So you can use many different ways to program it. So these GPIOs you can use to program certain filters.
So I think there's no more questions. So we can conclude this session.